Welcome to the stream. It's been a long time, about a week, and I know that we are definitely past when I was trying to do this stream, but oh goodness. I had to wait on some flow cells in order to do the sequencing that we're doing today. So, yes, welcome. Oh goodness. Yeah, I should have checked and made sure that my microphone wasn't muted. I had been going through my checks and I obviously forgot to go through that one. Excuse me. But yes, welcome to the bench. We're going to be doing some long read DNA sequencing today, which is different from my past streams. Past streams were all about cloning, which is an older molecular technique. We're going to be doing some more modern technique, which is long read sequencing. But yeah, I hope you're having a good weekend. I hope you're having a good day, morning, afternoon, night, wherever you are. Thank you for joining me. I'm just going to say what I had already said all over again, now that you can actually hear me. It's going to be a longer stream today, probably around 4.5 hours, just because this is a long protocol that we're going through, and somewhat new protocol as well. I got a new kit that we're going to be using today, so we get to go through that together. It's not terribly different from what I've done before, but it is new. We have Bert and Ernie joining us today, which is our double-stranded DNA mascot. So he's joining us because we're going to be doing the long-read sequencing. So yes, I hope everyone's doing well, now that you can actually hear me. And thank you for letting me know that you could not hear me. I appreciate that. Yes, thank you. But yeah, so yes, thank you for joining me. And I haven't recording this, so I was gonna say, because this is a long protocol, if you need to duck out at any time and you feel like you're gonna miss something, don't worry. I'm going to be putting this up on YouTube once the day is over, so you can always check it out on my YouTube channel, which is here. So if you missed the stream, you can always catch up. If you missed the cloning streams that I previously did, you can catch up on those. But yeah, so we're going to be starting basically a brand new technique that we do in the lab today, which is long-read sequencing. So these words obviously don't make any sense right here, but long-read sequencing, I was just discussing what the difference is. And this right here, so we have been able to sequence well over 200,000 base pairs, or if you hear me say 200 kb. That's what I mean. That is 200,000 base pairs. Hopefully you can read that well. So this is what I mean by long read sequencing. And this is, so short read, if you wanted, the opposite would be short read sequencing. And this is what most people might be familiar with. If you know about sequencing, there's Illumina. That's like the biggest company that does a lot of sequencing, and that's I think, oh gosh, where are they sitting at? 500 base pairs. I could be off on that. They might be able to do a little bit longer than that, but they are nowhere near 200,000 base pairs. They are doing very short fragments of DNA. So we are doing long read sequencing, and the platform I'm going to be using is called Oxford Nanopore. So this is a relatively new company that has come out, and we're using their kits, their sequencers for this. Another company that does long read sequencing is Specific Biosciences, or PacBio. They've been in long read sequencing for much longer, but it's very expensive. This is not a sponsored stream, by the way. I'm just saying. I'm just letting you all know what we're talking about. So Oxford Nanopore and PacBio are going to be the two companies that do long read sequencing. Now, why do we care about long read sequencing versus short read sequencing? Well, the big thing is with long read sequencing, you're going to get long pieces of DNA, long stretches of double-stranded DNA. And sequencing means that we're going to be able to look at all, this is just random base pairs that I'm putting up here, all the base pairs in this sequence without breaks. So it's etc. So this is long read. I'm just going to do long read. We'll do that. Long read sequencing. Short read sequencing, this is where you get very short fragments of the same thing. Don't cut out on me, purple. And then it's gonna break, and we'll have continue, break, continue, break, continue, break. So instead of having one just continuous sequence, we're gonna have short fragments broken up within this entire sequence. And hopefully it's not too, get a little bit shiny. Let's see. Is it better if I turn the light off right now? Oh, there we go. It's a lot better with the light off. Not too terrible. Yeah, the lighting can be a little wonky for trying to do this. And unfortunately my purple's starting to wear out on me, so let me get a different color. Purple and red, we'll do some red. Red sounds good. So why do we care about long read sequencing versus short read? So we want to have long continuous without any breaks. That's great. Because if we're looking at a specific strand, which is what we're going to be doing today, it's better if we are able to get this long continuous strand because we want to be able to see everything around what we're looking at. So basically, when you're going and you're going to sequence this thing, and then we want to look for something specific. Say we're going to do this. So we're going to do an enrichment today. Basically, we're going to try to pull out strands that have a specific sequence that we're looking at. So that's what this enrichment is that I'm talking about. So that's basically the two techniques we're doing today. We're going to enrich for a specific sequence of DNA within a genome, and we're going to sequence it. So those are the two methods we're doing today. And the enrichment protocol that I'm going to show today, we actually have published in the lab. So you get to watch as I do a published technique for you, which is always nice. And in case people find the paper and they want to look at it, they can always come back. But yeah, so we're going to enrich for the specific sequence, and what's really important is we want to be able to capture this entire sequence and everything around it because it's extremely important information versus trying to capture it in a short read. We're trying to capture this sequence in here. We're not going to get anything over here, and we're not going to get anything past here. So it's nice to have the long read in order to see everything around it. We want to see basically the sequence landscape around it to be able to look at the differences in the sequence compared to a short read one. And it's also easier when you're going through the sequencer for the actual downstream analysis. Because what happens is after you're sequencing, you're going to have lots of fragments of the same thing. They're going to pile up, and it's just easier for downstream bioinformatics. I'm not that well versed in the bioinformatics part of this, so if you have any questions about that, that will be a little bit harder. I'm not so good at that because I'm not into the programming aspect of the lab. I just do the nice bench work. So if you have any questions, let me know. This is the first time I'm kind of going through this in a teaching moment for people. So if you have any questions, please let me know. Especially if you're like, I don't understand a single thing that you just said. But yeah, so it's going to be a lengthy protocol that we're going to go to today. But it'll be nice to see it from beginning to end. And I unfortunately won't be able to raid the channel that we usually use to show the sequencing because, oh goodness, there was an update on OS and we don't have permissions in order to get into OBS for some reason, and it's just not going to work. So unfortunately I won't be able to show that. But this isn't the last time I'm going to be doing sequencing experiments. Hopefully we'll get that fixed for the next one, and you'll be able to see that as we go through it. But at least you'll be able to see from beginning to end. All right. And also, we've got some nice incubation periods, and if you had joined me for my previous cloning, the very last stream that we did, I was going through and showed you a gel that we ran of the plasmids that we made. I'm going to go through that with you guys today, so you have an idea of what to look at when you're analyzing a gel. And we've got plenty of time to do that in the incubation steps. All right, so hopefully this was helpful. This little diagram was helpful. We can always go through it again later, not a problem. But I'm going to get started so that we don't lose too much time in the beginning. So let me just put this away so it doesn't get dirty. say goodbye to Bert and Ernie. They're gonna head off in my markers. Alright, so now I'm gonna turn my light back on so it's better. There we go. Now, before we get started, PPE, of course. I've got my safety glasses on. So we're all set for the first set of PPE. Second, lab coats. Don my lab coats. Get it honest, of course I've got my purple lab coat. And then gloves. Gotta get our gloves on. There. And we'll be all set. We've got our PPE for the day. Okay. So, first thing we have to do when we're doing this setup for the enrichment that I was talking about, so specifically targeting a sequence in our genome, is we need to put the DNA into a PCR tube. Very important. And the enrichment that we're doing, we're actually going to be using a protein called Cas9. I'm just getting set up here. And Cas9, if you're familiar with CRISPR, is the protein involved in CRISPR. We're not doing CRISPR. CRISPR is usually when you're actually going in and modifying a genome using the protein. We're not doing that. We're using it to enrich for the specific DNA sequence. And what's nice about this protein is you basically just give it a sequence, like its own little short sequence called an sgRNA that matches what you're looking for. It's about 20 base pairs of the sequence you're looking for. You give it to the protein. It will combine with that sgRNA and make a complex, so a protein complex. And using that little sequence that you just gave it, it will find that sequence in the genome and it will cut at that sequence. And that's what we want to do. That's just how we're going to enrich it. It's going to cut at those specific sequences. And sorry, I'm just trying to label my tubes. So we have those all set and ready. So it'll cut at the specific sequence, and by cutting it, we're going to be able to add sequencing adapters to it. I'll talk about it more as we go through. I'm just trying to give you an overview. And so the point is that by adding the adding the adapters to it, so the sequencing adapters, when we put it on the flow cell for sequencing, we use flow cells when we're putting it on a sequencer. We're going to put it on the sequencer. It'll find those pieces that have the adapters ligated to it. So ligation, if you remember, is when we add something. We're going to add something to the end of the DNA, and it will sequence those specifically. Hopefully, that's the plan. That's usually how it works. And that's what Cas9 does. It looks for that specific DNA for us, cuts it, and allows us to put those adapters to the on the end of it. All right, so these tubes right here, I'm using two different PCR sets because one of them is for the genomic DNA that we're going to be using. So this is already extracted genomic DNA. The other one is for setting up the protein complex with the Cas9 protein. And one of them has to sit at room temperature, which is the Cas9 protein. The other one has to go into our PCR machine. So what I'm going to do right now is some quick math of how much I'm going to use of our DNA. And I'm going to be using, so this number might not be familiar to you guys. We're going to be using a total of 10 micrograms, which is basically just a measurement of how much of the DNA we're using. So micrograms, think of it as grams. You've heard of grams. It's just micrograms. Very small. Very, very small. Micrograms. So we're going to be using 10 micrograms of the genomic DNA. And then add some water, put it in with some buffer. And then what we're going to be doing is the DNA will sometimes break even if it's just sitting around. And we don't want the Cas9 to, we don't want any adapters ligated to that as much as possible. We're not going to get 100%. So what we're going to do is called dephosphorylation. So we're going to add an enzyme that will take out a phosphate that's sitting on the end of the DNA. I know we're getting into some chemistry. I'm going to bring over Bert and Ernie. So Bert and Ernie are broken, as you can see. And when they're broken, there's going to be a phosphate that's broken here. So basically that free end, that free phosphate, if you add the protein called ligase, which is what we use to put the adapter on, it's going to add something to it. It finds that phosphate, is like, oh, this is where I need to add something. All right, let's stick it there. Well, we don't want it to just do that willy-nilly. We want it to do that with the specific sequence that we're looking for. So in order to get rid of any phosphates that could be floating around in the DNA, because DNA breaks just if you handle it, we're going to dephosphorylate it. So what it's going to do is this enzyme called CIP, is going to come in and it's going to be like, oh, free phosphate, get rid of it. Get rid of it. So they'll get rid of that phosphate and it's gone. The ligase, which adds things, won't see it, and it won't add the adapter that we're talking about. So that's the first thing we do. We're going to get rid of any of these phosphates that could be sitting around, and hopefully we won't sequence those. So I'm just getting that ready. So we're going to need to add buffer, the genomic DNA, the protein, which is called CIP, and water. Excuse me while I do a little bit of math, just so I know how much water to add. I'm just writing in my lab notebook off to the side. There we go. Okay, so I have that all set. I've already got our buffer that we need and our genomic DNA. So let's do that first. Well, actually, I need to go grab some things from the freezer first. While I do that, I'm going to grab a specific buffer for the Cas9 protein, the sequence that we need in order to do the enrichment. Grab that, and the Cas9 itself as well. Let's see, we got it here. Not the this one. Let's see. Yes. Here it is. Okay. Grab the things I need, and of course the CIP protein. We need that as well. There. We're all set. Okay. Now we have it in our handy dandy little freezer caddy that keeps everything cold when we're going to use it, and that's our specific protein that we're going to use. Alright, so let's add the water first. Always add the water first, because if you mess up on that, it's very easy to fix. I need 74 water. Water right down into the bowl. Go. And then I need 10 microliters buffer. Those are making up to 100 total. Go. I've got the buffer right here. Mix it a little bit. Alright. And then we're going to need 10 microliters of the genomic DNA. I'm using a bit bigger tip because we don't want to shear our genomic DNA. So what I do is I hand make a wider, more tip by cutting the end of it with a razor blade. So the end of it's a little bit bigger, because the smaller the tip end is, the more it's going to break up our genomic DNA. Alright, here's our genomic DNA. Alright, let's see if you guys will be able to see this. Here, I think I'm going to move it just a little bit so you guys can see when I'm talking about viscous DNA. Hopefully you're going to be able to see it. it's actually stringy. Oh, you couldn't see it very well. But when I was pipetting it up, it's actually very stringy. So it's very viscous DNA, which means we have a lot of DNA in there. A lot of DNA. Alright, so that's the genomic DNA. Now I need to add the SIP, the protein that's going to get rid of those phosphates, get rid of those ends that we don't want it to ligate to. At 6 microliters. And we use something called quick SIP because you can heat and inactivate it. Inactivate it. There we go. All right, that's added. Now I mix this way because we don't want to vortex. If we vortex, it's going to shear our DNA, and we want to have nice long ends. All right, I have to go spin this down and then go put it in the PCR machine. So you guys hang out. All right. So I just put the genomic DNA with the SIP. It's going to incubate at 37 degrees Celsius for 30 minutes, and then quick 80 degrees Celsius to kill that SIP protein, and then we're all set. So while we're doing that, I'm going to actually put our genomic DNA away. That needs to go back into our fridge. We're going to make the Cas9 complex that I was just talking about. So what we're going to be doing is adding the Cas9 protein to basically the sequence that we're going to be targeting for. So that 20 base pairs of the sequence that we're going to be targeting for. So it's just a small part of the sequence that we're looking for, but it will be able to pull out the larger chunk because it matches. But we need to have the protein and that, like, it's the sgRNA that I talked about, come together. So they need to form a complex. So that's what we're going to do right now. And I need to do some quick math on that as well, because I need to figure out how much of our sequence we need to add. So, and once again, we're going to be adding buffer to it, water, of course. As always, we usually add it all up to the same amount. Let's look. I know I checked it. I made it, and I know I added it. Where did I put it? I just reused it. Oh, there it is. Looking back in my lab notebook. Always to check. Always to check. So I write everything down in my lab notebook. Alright, perfect. Okay. So, let's see. First thing, always, we're going to add water. 9.6 microliters. I'm using a different water just because I'm a little paranoid. This is definitely RNase-free water. Because sgRNA, if you heard in the name, is an actual RNA. which is different from DNA, and that it's missing. So DNA is dideoxy, RNA is just ribonucleic acid. So, no, deoxyribonucleic acid. Sorry, it's a Monday. I can tell you can already tell it's a Monday. DNA is different, and RNA is ribo. So it doesn't have the Actually, oh my goodness, I can't think today. Excuse me, I can't think today. Anyway, RNA is less stable than DNA, and is usually not fun to work with. So I always make sure that there's RNA-free water whenever I use it. Other people have used it regular water. This is just me. All right, so we added the water. Now let's add the buffer that we're gonna need. We need 1.5 microliters of the buffer. Alright, so the buffer is set and ready. Now we need to make up our Cas9 dilution. So we actually dilute the Cas9 because when we buy it from the company, it's a little too high concentration. So we actually dilute it out. So we have a certain ratio of protein to the sgRNA that I'm talking about. This is the one I want. I need 2 microliters. And we dilute it in basically this, but a diluted form of this buffer. I know, a lot of dilutions. I just said a lot of dilutions. And that's what this is. I just made up a dilution of that other buffer, so it's ready to go. Makes my life easier. We just add it to this one. So this is where we're going to dilute the protein. Right there. And let's grab the protein. So we buy the Cas9 protein from a company that makes it for us. Oh gosh, is there anything in here? Yes, there we go. Yes, I got it. And then I'm gonna take this out because it's off. And so it's a very small amount, 0.5 microliters. Right down into the buffer. And some of it came up, so you know what I do whenever something like that happens. I take off the tip and I put it back in, and it usually forces it out of that tip. Right. And then I can flick to mix this one, because this is okay. We don't have our DNA. And then I use a microliter of the diluted protein in our mix here. Just tap this down. And let's grab that protein. So it's a very small amount, if you guys can see. Very tiny amount of volume that we're working with here. 1 microliter. We're good on that. I'm going to put it in here. Our water buffer and protein. And now we just need to add the sgRNA. That is our specific sequence that we're looking for. And I need 2.9 microliters. Right there, 2.9. But it's got to thaw. Remember, 3 things that we do in the lab. Wait for centrifuge to finish. Wait for something to thaw. And, oh gosh, see, it's a Monday. What else what's the third thing that we're always waiting for? That we're always doing? Wait for a centrifuge. Wait for things to thaw. I mean, wait for timers. Thanks captioning. I saw that timer. Oh, it's so a Monday. I can't remember the third thing. Oh, you're seeing me on a very tired Monday, everyone. A very tired Monday. Oh, there we go. Now it's almost done. It's almost done. You can see. Almost. There we go. All set and ready. All right. I'm just gonna fit to mix it just in case. Tap it down. Then we're gonna add that. Our mix here. There we go. And then do a little bit of this. for mixing. And then I'm going to spin it down briefly. Oops. And then put everything back in the fridge. So you guys get to hang out again with the music for a little bit. All right, I'm back. Okay, so basically everything is incubating right now. We've got 27 minutes for the dephosphorylation of our genomic DNA. I'm going to bring this over here. RMP is just going to basically sit here until we're ready to use it, which is our next step. We're going to add the Cas9 with the sgRNA. Complex is called RNP. I forget what it stands for. We're going to add that to our genomic DNA, and then that's when the protein's going to go through, the Cas9 is going to go through, and it's going to cut the specific sequences that we're looking for. So that's the next step. But right now, we're just hanging out. We're just going to hang out. So I'm going to take off my gloves for right now, because we're all set for that. All right. So, I mean, yeah, was number 3 wait for a Geltron? Tree Wizard? Oh gosh, I can't remember. Oh, I can't remember what I said. What is it? Wait for things to thaw. Wait for a centrifuge. Oh, and move colorless liquids from one place to another. That's what it is. But yes, wait for a gel to run. That could be number 4. We can make that number 4. Yeah, I didn't sleep very well last night, everyone, so I'm tired today. And of course, it's a Monday. So you'll have to grant me some mercy today while I go through my teachings. But I'm just glad that I could finally do this stream for you all. I'm so happy that I had to wait. I'm happy that I get to do it. Not happy that I had to wait for the flow cells to come in. And ooh, you know what? I think I'll show you what I was talking about so you all have an idea of the thing I was waiting for. Yeah, so in order to do the sequencing that we're doing today, we have to use specific flow cells. And basically this is what it looks like. Yay! So we're going to be adding our enriched DNA to this at the very end. But I had to wait for these to arrive in the mail in order to do this today. So yeah, so towards the end we're going to be using these. So flow cell basically, I mean this is specific to Oxford Nanopore, so the company that we're using. This is what they look like. I don't know what PacBio's flow cells look like or how that procedure goes. I've never done PacBio sequencing before. So don't ask me, I'm not sure what they look like. But this is what we use. And on this, it's going to go through and it will sequence our genomic DNA. So it's basically right here where this yellow spot is where the pores are in the flow cell. And the pore is where the sequencing happens. It grabs a piece of DNA and it goes through basically another protein that's sitting on here. And as it goes through, it's going to check like electrical signal changes. And that's how it decides what a base is. It's pretty cool. If you search online for nanopore sequencing, it'll give you a cool like YouTube video about how the actual sequencing works on one of these. But yeah, so we're going to be using this later today. So this is the culprit. This is the culprit. This is what I had to wait for in order to do the stream today. So we're all set. We're all set on that. But yeah, so now that we're hanging out, does anybody have any other questions? What are we doing today for all of you that might have stopped in? I can let you know what we're doing. So we're looking for specific pieces of DNA. So we're doing an enrichment in the genome, and we're using long read sequencing. I'm gonna bring my thing back over today. This is what I discussed earlier. So we're doing long read sequencing. Long read sequencing is basically looking at long pieces of DNA. And personally in the lab, we have sequenced well over a piece of DNA that is 200 kb or 200,000 base pairs. It's very big. It's very long. Versus short read sequencing. So if you've heard of Illumina, that's short read sequencing. They are looking at something like around, I think it's bigger than this. Probably. I haven't done Illumina sequencing in a very long time. 500 base pairs. So very small. Very small compared to what the sequencing we're going to be doing today. And the company we use is Oxford Nanopore. This is not a sponsored stream. This is just what we use in the lab for doing this sequencing. Pacific Biosciences is the other long-read sequencing platform, but they're very expensive. Very expensive. But yeah, so why we're doing long-read sequencing, we're looking to enrich. This is just a diagram. I don't know the specific sequence off the top of my head. But what we want to do is we are looking for a very specific sequence in the genome, and we're going to be using a protein called Cas9, which if you're familiar with CRISPR or have heard of CRISPR, that's the protein involved in CRISPR. And what it's going to do is going to find the specific spot, the specific sequence, and it's going to cut at that spot. And basically what that cut does, is it's going to allow us to put an adapter for sequencing and add it to that. And so we're going to be sequencing up the specific DNA. And we want the long read, we want this to be longer because we want to look at the sequences around it. Short read, it's basically going to be like, oh, it's going to stop. We're not going to be able to get much of the genomic landscape or the sequence that's around it because it's going to be chopped up pretty fine in that case. And there's other bioinformatic reasons around that too for the analysis as why long read is nice versus a short read. I'm not as well versed in the bioinformatic analysis downstream, but this is important as far as at the bench we want this long read. So if you have any questions, please let me know. Please let me know. Ooh. But yeah, so if you have any questions about the Cas9 or how that works, I'm open for questions as always. And then yeah, so we're just waiting for the dephosphorylation step to finish. I think we're at 20 minutes now. Yeah, 20 minutes. Look at that. I guessed right. 20 minutes, that'll be set. We've got our Cas9 complexing with the short little sequence I was talking about, the sgRNA, that's what it's called. It's doing its thing right now. And then we'll get that set. But I hope everyone is having a great day today. It is definitely a Monday for me, but not terrible. I get to spend it with all of you doing some bench work. So if any of you have missed the beginning of the stream and you want to check it out, this VOD will be up on my YouTube channel later. So if you missed it or you missed anything else like today, this is a long protocol. It's going to be another 3.5 hours. So if you have to duck out, it's all good. You can always check the YouTube channel later, or this VOD will be up on Twitch for a couple days as well. So if you want to go back. And just another plug for my YouTube channel, just in case you want to find my other VODs, you can go there. Basically, the other streams that I did was going through cloning, which is an old molecular technique from the 70s that we practice here in the lab. But today we're doing more modern stuff. We're doing actual long read sequencing. So this is basically my main project is going to be the sequencing. So if you just miss it, you don't want to catch the VOD, I'll be doing plenty more of these streams. Don't you worry. Don't you worry. You're not going to miss too much. But one of the things that we did in my last stream was we we're looking at a gel, and I promised that we would do a gel analysis of what we were looking at. So let me pull up the gel for you guys. So these are going to be gels of, there. Hopefully you can see it. Hopefully you can see it. So that was one of the gel pictures that I took from the last stream. And this is based on, look, I even printed it out so we can look at it together. I'm gonna move you over to the side here. There. So we can go through it together. So we can analyze this gel together. So basically, what we want to do is look at this gel and see if the plasmids we had made previously, it's gonna be a lot easier if I turn my light off, there, are what we want. So this is the second gel image, and I'm just gonna orient you right now. So the first lane, so I have two separate lanes, because we did two different restriction enzyme digests, or we cut the DNA specifically into fragments with using two different sets. So that's what these are. This is our molecular ladder. So a molecular ladder is just DNA that is cut at specific sizes. So, and this one, this is 100 base pairs down here, lower band, all the way up to 15 kb or 15,000. So this is a very top band right here is 15,000. And this one, just to orient you, this band is 1.5 kb, 1,500. This is 2 kb, and this is 3 kb, 4, 5, et cetera, et cetera. This top band right here is 1,000 base pairs. Then it goes to, it's not exactly 900, it's actually 850 base pairs of the next one. And I have a nice little cheat over at the side because we use this ladder a lot. And 650, and then from there it goes to, this is 500, 400, 300, 200. So that's how I'm just orienting you as to what we look like, what we look for when we see the molecular ladder here. And then this was our uncut plasmid. So we didn't use an enzyme to digest it out. We didn't use an enzyme to cut it. So this is what a plasmid looks like if you run it out on a gel without cutting it. So basically it's circular with maybe some cuts in it. So this is circular DNA. Circular DNA, that's a circle. This is a circle. Because plasmids are circles. And then these guys, I've got my handy dandy gel sitting here. These are all the same plasmid that we made. So this is 1, 2, 3, 4, 7, 8. So 1, 2, all the same. These are the same plasmids. Difference is we use different enzymes, different proteins to cut them. So I'm sure you don't remember, or this is the first time you're here. I'm just gonna put the names of the enzymes we used at the top. So we used DRAW3 and AVA1 at the top. And on the bottom here, we used EXPA1 and I call it NICO1. So there's two different enzyme sets. Now the reason is we want to be able to see if we get the correct plasmid by looking at what it's supposed to look like. So this is what it's supposed to look like. If the fragments are correct, we want it to look like specifically this one for this one, this one for this one. And of course the ones on here, so this is what we're looking for, just so you know what we're talking about. So one thing is there could be 3 specific possibilities that I can think of when it comes to doing the plasmid cloning. One is so we're trying to put a gene into this plasmid. We were basically inserting a gene into a plasmid so that for downstream stuff, I need this gene. One thing is the vector, which is basically the plasmid that we're trying to put the gene into, just religates on itself. It just comes back together. It's possible. It does that all the time. And that's what this banding pattern would look like. If it's the plasmid we want, this is the pattern we're looking for. So we're looking for basically a band between 3,000 and 2,000 kb, one at about 500, and another about 200. Now, one thing is the gene could have gone in forwards or backwards based on this cloning scheme. We want it to be forward. It will not work if it inserts backwards. So that's why I have backwards with a sad face, because we don't want this banding pattern. So that's what this one will look like right here. So the difference between these two is this is slightly higher, closer to 3,000. That's kind of hard to see on a gel sometimes. But the big one is down here. If it's forwards, we'll have a band at around 500. If it's backwards, it's going to not have that 500 band. It'll have one sitting right basically around 350, and then a lower band. So that's with, I can't remember which one it was. Which one was it? Oh, this one. So it's slightly different from what we're going to see on here just because of the way I set it up. This is going to be for the neat. Oh, my band-aids falling off. Sorry, we're fostering some kittens, and one of them was a little aggressive while I was trying not to let him get out of the pen this morning, and he scratched me up. That's why I had that band-aid, but it fell off. And then this one is going to be our draw 3, maybe 1. There. This is just the way, and that's why we have the 2 sets, so we know which ones to look for. So we did the 2 because if I just did the draw 3 and the AVA 1, I can't tell the difference between a forward and a backward plasmid. All I know is that the gene got inserted. So that's why we do a second one to see if it's forward and backward. We know the gene's there, but we need to know if it went in a forward orientation or a backward orientation. So that's why we did the 2 different ones. So, looking at our gel, if I can pull it up here, let's look at which is the first one? We got the top one. Let's look at the top one and see what we got. So basically, you just look at the gel, look at what you are expecting, and see if it matches up. So if we look at number 1, we're looking for about 500 and around 400. Oh, see, these are lower. And when the top band should be around 250. Top band looks right, but these 2 bands right here are sitting more at 200 and 300. I just want to double check. I did print out another one because we wanted to see it. Yeah. So 200 and 300. That's not what we're looking for. That's not what we want. That's not what we want. We want a little bit higher than that. So, like I said, funky things can happen. Definitely. Funky things can happen when it comes to cloning. So it's possible that we got just some weird stuff occurring. Yeah, these are, unfortunately, so this one's bad. This one, nope, bad. Alright, so we have one at 500 and 200. Oh no, that's something that we didn't even see. That one's bad too. That's not right. And it's possible we don't get a positive. It happens. This happens all the time. It just means I have to go back and redo the cloning again, which is okay. Alright, and then... did I do this one right? Sorry, folks. I need to make sure that I'm looking at the correct version of this in here. Because it happens all the time. I have like 5 million notes. I want to make sure that's the correct notes that I'm looking at. Expa Nico. Oh, I have this one. Haha. That's why. Yep, this is why I look at my notes. It's actually flipped. Oh, it's gonna be a fun day today. Welcome to the lab. Happens all the time. Like I said, doesn't help that I'm tired. So this is actually draw 3, and this is Expo. Switch! Switch! Oh, we're looking at the wrong one. It's a fun Monday. Alright, let me double check that our stuff is okay. Alright, we got 9 minutes until the incubation's done. Alright, let's reorient ourselves on this one. Doesn't change what I said before, it just changes which part of the gel we're looking at right now. So actually, down here, this isn't the best picture in order to show the Expa and the Nico one, so let's move it over. Because the other one, let's pull it up here. I have the other one. Let's see. Is it this one? There we go, there we go. That one's better. This one's better for showing the Expa and Nico. There we go. Yeah, there we go. Much better. And then, same kind of thing. So this will be the same one. Same exact setup as the one up here. So I'll put it on here. I'm just going to call this ladder. And then uncut plasmid. And then 3. Yeah, so down here, it's a lot easier to see the Expa and Nico because we can see it closer down here. That's what it is. Because we're looking on, when it's this small, they're going to look like just a wide band here because they're so close together. So, all right, back to what we're talking about. We're looking at just these 3. Here we go. So we want to be able to see the difference if there's a band sitting here at about, I think that one would be about 600, so that'll be around 600 base pairs versus basically a double band sitting around 500, 400 base pairs. So we want to be able to see this one, not this one. So if we go through here, top one is still going to be the same. It's going to be at, I have the exact number, 2.7. Nope, 2.4. 2.4 kb. So this is the 2.4 kb is what we're looking for. And then a double band, so this 100, 200, 300, 400, 500 base pairs. So double band sitting around here. So all of these right here look good. Check. This one, not so much. This one's got the 2.4. It's got a band sitting at where we want to be. It's got this extra band. No idea what this extra band is. What is this? And it's not at 600 because that would be here. So it's higher than 600. So I don't know what that is. That's funky. So we can say nope, which means that 8 up here is also bad. So we know that the gene inserted on these guys. Yay! Yay! This is a thumbs up. Thumbs up. But we don't know if it inserted forward or reverse in this case. Yeah, there we go. So that's why we look at, I mean, we can see it here kind of, but it's easier to see it on the other one because it's been run out a little further. But we can look at it from this point of view. So let's do this. There. So the big ones that we care about, right down here. Right here. So we want to see if it's inserted forward, we want a 500 band. And we want one at 200. If it's reversed, it's going to be at about 350 and 200. So 100, 200, 300, 300, 400, 100. This is why I like to run it out a little more because it's hard. These bands are like close together. But we should be able to see the difference between a 500 200 and a 300 200 because 300 200 is going to be much closer together than the 500 and 200. So for the first one, yeah, we've got what looks like a 300 200. So I'm betting this is a backwards one, and we'll look at the other gel to confirm it. But all good, and we already know that 8's bad, so don't look at 8. For number 2, looks like we've got a 500 and a 200. So number 2 looks great. This means that it's probably forward. Yay! So basically, if we think what we've got, this one could be correct, and this one is bad. Alright, so 3 looks like it's probably backwards. This one got funky. We got some extra bands here. What's this extra band? What's this extra band? What's this extra band? These guys have extra bands. And this one, number 5, looks like it might be, although it's hard to see. We can't see a 200 on there, on that one. But I'm already going to X out 4, 6, and 7. Because we have extra bands here that I was not expecting. We don't know what that is. So we can already knock out 4, 6, 7, and 8 because we've got some weird results on that one. So let's look at this one up here to see if we are correct and we get 1 or 200, and that is this, what we were looking at before. So we know that 4 is bad. We know 6, 7, and 8 bad. So we want to look at, and I'm guessing that number 2 is probably backwards. I'm just going to call it backwards. But 1, or sorry, not number 2. Backwards. Number 2 looks like it's probably forwards. Number 3 looks like it's backwards. And number 5 might be forward, although I can't see the 200 below. I can just see the 500 and the upper band. So if I really wanted to check number 5, I'll probably have to redo that one just to make sure that that one's there. But what we really care about is if 1, 2, and 3. So we want, so this is 200, 200, 400. So remember, we want 500, 200 if it's forward. So number 2. 500, nice. 200, great. Number 2, looks like it's our candidate, the one that we want. Now looking at number 1, that one would be, if it's backwards like we suspect it to be, we'll have a 300 to 200. Look at that, 300, 200. Backwards, it's really light, but you can see it here. 300, 200. Backwards. So we can't do 1 and 3. And then 5, we just completely lost it because it ran out too far. So if I want to check 5 and see if I've got another forward, I can do that again. But number 2 looks great. This is what we wanted. This means that our cloning that I did with you all on stream worked. We got 1. I already told you that there's not 100% efficiency. I wasn't expecting that, especially with the cloning that I did. This was difficult sequences to work with. 1 out of 8 ain't bad, possibly 2 out of 8. So I'm okay with that. Yay! I'm so excited. I'm so excited it worked. Now, if you have any questions about what I'm going through, feel free and ask. If you want to catch up on this whole thing from the beginning end, we actually made these plasmids on stream. We went through and transformed them into E. coli, grew them up, did the DNA extraction, and basically digested these for analysis. So we did them all together on stream. And those are all up on my YouTube channel if you want to go back and look at that. But yeah, so this is basically what we do once we do some cloning to check if what we got for a plasmid is correct. The next step would be to actually sequence it, make sure that everything is there. I mean, the digest looks good, but small things you won't be able to catch on a digest. That's why we will also go through and sequence it. And that's probably going to be done on stream at a later time when I have more plasmids to sequence, because we're going to do long read sequencing on plasmids. And in order to make it cost effective, we have to have a lot of plasmids on there. So 1 is not cost effective in that case. But I'm so glad. Number two. But I'm going to put this away. But if you have any questions about that, feel free to ask. I don't mind going through it again. But yeah, so awesome. I'm so glad we got to go through that together. And the gel looks much better than it did on my phone. Much better than it did on my phone. All right, perfect. Our incubation is done. I'm going to turn the light back on so it's easier to see while I pipette. All right, so the next step in our enrichment and sequencing protocol, I'm going to grab our DNA out of the PCR machine and then set it up for the next step. Second here. Sometimes our PCR machine takes a while to. There's it. No. Where'd it go? That's fine. I'll set it up. Okay. So here's our DNA. What we're going to do is we're going to add the Cas9, and we're also at the same time going to add an A. So an A nucleotide at the end. And why do we do that? Well, the reason is that the sequence sequencing adapter that we're adding has a T overhang. And I will write this out for you guys. So I'll draw it out so you can see it. So so it doesn't take just the Cas9 cut. We have that phosphate there, but it needs this A in order for it to actually ligate the adapter on. So when Cas9 cuts, another protein's going to come in, and it's polymerase. So if any of you were familiar with a polymerase basically just makes copies of DNA for doing a polymerase chain reaction. We're going to use that to add A's. It basically goes through and adds nucleotides to a DNA strand. That's what it does. That's what a polymerase does. So it's going to, if we just give it A's, when it sees that something has been cut and it has that free end, it's going to add an A to it from the Cas9. So we're basically doing the Cas9 cut and adding A's in the exact same tube at this point. But let me double check my math on that as to how much to add. Should be 15, so we got 15 of the RNP. We've got 100 of our gDNA. I'm just writing this down for later. Since I need it just in case. It's going to be 1 microliter of the A nucleotide, 1.5 microliters of our Taq polymerase, and Taq is just the name of the protein, the specific polymerase. 15 microliters of our RNP. And what does that leave us at? Let's see. $117.5. Sorry, I just need to do that math so I have it for later because I'm going to need that for the next step. Okay, so I'm going to add our RNP first. So it's 50 microliters. That's what we made it up to. So take it straight out of this tube. And then we're going to add it to the top. We're not going to mix it. We're just going to add it straight to the top. Because remember, if we mix right now with our pipette, it's going to shear our genomic DNA. We don't want that. We want to have nice long fragments for the sequencing. And then I'm going to add the ATP. Sorry, not ATP. The ATP. So the A nucleotide. Same, right at the top. Then we'll add our 1.5 microliters of Taq polymerase. That's right here. Okay. There. Add it to the top. And then we mix it. Just like this. So no vortexing, no pipette mixing. We want to keep those fragments long. So any kind of vortexing, any kind of mixing with a pipette is going to shear our DNA and make it into smaller fragments. This is an easier way to mix it without losing those long fragments. All right, I'm going to go spin this down and then put it in the PCR machine, and it'll be at half an hour at 37 degrees Celsius so the Cas9 can do its thing, and then 75 degrees Celsius for the Taq polymerase to add the ACE. So that's going to be the next one. So it's another long incubation for us. But I'm going to go do that right now, get that started. I'll be right back. All right, so I am back. Okay, so since we have a nice long incubation, I'm going to take a quick break right now. You guys can also as well get something to drink. Go to the bathroom if you need to, grab a snack. I also need to grab some of our other solutions and some ice. So that'll take a little bit. But you guys can hang out with the music and we'll be back in about 10 minutes. Sound good? All right. See you back after the break. All right. We're back from break. Hope you guys were able to take a nice little break. I got something to drink just because I'm getting a little parched while I'm talking. But welcome back. I also grabbed some of the solutions that we're going to need in ice pit. Need an ice bucket for what we're going to continue with. So I have these sitting out because I need to thaw them. Remember, one of the things we do as scientists, wait for things to thaw. So these need to be at room thawed and then placed on ice. So these are just solutions that we need for the actual sequencing that we're going to be doing. And it comes from the kit that I'm going to be using. But yeah, so while we're waiting, we already went through our gel and the last incubation. What I'd like to go through right now with you all, I'm just going to move my stuff over, is basically what the Cas9 is doing. Let's see, what color do I want? I used red. Who do you use red? What? Red and black. Sure. We'll just do red and black. Then we get a new page for ourselves. Alright, so yes, so as I was saying, so today with the enrichment we're using the Cas9. I'm just going to diagram out what it actually does. So here we have our DNA, and I'm just going to do a sequence, just a random sequence that I can think off the top of my head. Yeah, and it goes on, and it goes on. And remember, this is double-stranded, so it's actually double-stranded DNA, so let's make it double-stranded. And if you don't remember how double-stranded works with DNA, A and T bond together, and C and G bond together. So if you see a T, there's an A on the opposite side. If you see a G, there's a C on the opposite side, and vice versa. So we made a nice double-stranded DNA. So this is our DS DNA. And our lovely mascot, remember, Bert and Ernie. Bert and Ernie is double-stranded DNA. Here we go. There he is. I wish they stood out a little better. They just crumpled together a little better. So, say we are enriching for, we're just going to say this sequence. This is a sequence we want to pull out. And we're going to be using Cas9 to do it. Ooh, let's get a different color for Cas9. Ooh, what's a good color? We'll just pull out the blue. The blue is working very well. So we want to get this. So what we add is we have our Cas9 protein. Oh gosh, no. 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 I don't want it to be that color. I don't want it to be like that. We said no. There. So our Cas9, we'll just make it a blob. He's a blob. Here's our Cas9 right here. This is Cas9. Right here. And we give him a sequence of, I forget which side, but it's going to be, we'll say it's going to, we want this sequence, so it's going to be, so basically it's going to stuck in there. So this is our Cas9 protein. We'll color him in. So we basically made this complex where it's the Cas9 and this little sequence we're looking for right here. And the sequence we actually use is 20 base pairs. So this is only 4 right here. But this is just an example of what we're doing. So what happens is the Cas9 actually will read through the DNA. It's actually going to sit on the DNA until it finds the sequence. So when it does, get it here, and it's actually like spitting it out as it goes. This is fun. I've never had to draw this before, so you get to see how much fun it is. We'll do this way because it's actually going along. So Cas9 is going to find this spot, because remember it's got G, D, D, G. So it finds that spot, And it actually is, so it's kind of like, like I said, I've never had to draw this before. Oh, see, this is why I gotta use water. Can't just do dry on this book. Gotta use water for it. So it actually erases it better. Oh, I got it. There it is. See if I can make this a little better. With my horrible, awesome drawing skills. My horribly awesome drawing skills. When it comes to science, there's actually like, so it's kind of more like a bubble instead of just all of it going out in the same. It's going to actually cut right here. So it's going to cut. It's going to make a cut for us. And what happens is, or this way, we'll have and yeah, it's going to cut on the other side. So there's actually a cut here, though it doesn't continue. It continues it this way, continues out this way. Same with this one. But here, we have our cut. And there's going to be like a little phosphate. 5' phosphate. So this little P right here is a phosphate. So this is what I was talking about with the cloning. And this is how ligation works, is you're going to use a separate protein that's going to do a ligation. So what we care about is like, this is nice and cut. This is going to head off in the other direction. It's gone. It's no longer a part of this. So it's a bye-bye. It goes floating off. Goes off. Don't care about that. Well, actually you do. I mean, there's going to be another phosphate here. In that case. Just because I made that cut. So, this is what the Cas9 is going to do. So it's going to look specifically for this sequence in the genome, and it's going to cut where that sequence is, leaving these lovely 5' phosphates right here on these sequences for us. So, the next step is when we add the polymerase that I was talking about. Now, the adapter actually, the sequencing adapter, which is what we need in order for it to be on the flow cell, in order for the sequencer to actually find these pieces of, oh, thanks for the follow, Pickle Lightbright. I'm so glad that you're following and you're enjoying the stream. Welcome to the lab. And I hope you can follow what I'm talking about. This is the first time I've had to describe this before and draw it out. So I hope I'm doing a good job. If not, just let me know and I can try to explain it better. Yeah, so the adapter won't be able to just sit on here because the adapter has a T overhang. And when I mean a T overhang, we're going to switch over to the next spot right here. Continue on. We'll just continue with our lovely one right here, G. A, T, E, C, G, G, C. With our little overhang phosphate. So we'll do the let's do the adapter in red. Here's our adapter. And I don't know what it is. I'm just going to make up a sequence for it. I have no idea what the adapter thing is. It's proprietary information, but I'm just making up a sequence for our adapter. So this is our sequencing adapter. Sequencing adapter. Oh, gotta have a T. Yeah, haha. Has this lovely T overhang that I'm talking about. All right. So it's double-stranded, just like thing, but see how it doesn't have a T here? No T right there. Or, sorry, no A right there. This is a T overhang. So this is important right here. So in order for this to be ligated onto our sequence that just cut from the Cas9, we need to add the A for this. So what polymerase does is it comes in and it's going to add that A for us. So polymerase, which is TAC, we call it TAC polymerase, adds this A for us. And so that will allow another protein to come in, and that's called ligase. We go from here, so now this is A. A. There's our A. And then our adapter. I have too many colors. Too many colors. There's our adapter. So this is our adapter. It's going to, ligase is going to come in and it's going to put these together. Basically going to add these together. So we'll make what ligase is going to come in and add that. And there's a lovely overhang here, so it's going to basically just click it together. So our final sequence, when we actually put it on the sequencer, is going to be our genomic piece that the Cas9 has come and cut for us. With the sequencing adapter. So it's going to look like this. Now this is just, like I said, a visual. This is just an example. I do not know what the sequence is off the top of my head at all. Okay, let me pull this up. There, a lot easier for you to see. So that's why we're doing these steps that I was talking about. So we're right now doing the Cas9, which is cutting at the sequence that we're looking for. Like, for example, this sequence. So it's doing this cut for us. And the tack is coming through. Sorry, I hit my microphone. The tack's coming through and adding those A's as it finds these cuts. So that's what's happening right now. The next step is to do this ligation with the adapter. So this is our next step after the Cas9 has gone through. Hopefully this makes sense. Hopefully this diagram is you can actually follow. If you have any questions, please feel free to put them in the chat. I don't mind at all. I'm just glad you're here and enjoying the science with me today. But yeah, so that's basically what's happening in a nutshell. And in a very, like, on paper kind of way when I'm describing it. So hopefully you understand what I'm talking about. If not, it's okay. And for all of the new people that have joined us today, welcome. Welcome to the lab, welcome to the bench. I'm glad you could join me. Hope you're having a good day, afternoon, evening, morning, wherever you are on Monday for me right now. And that you're enjoying what you're seeing right now. I'm just checking to see. I had some of these out thawing because we need these to be thawed. So yes, got stuff thawed. Looks like it. I need these thawed and then put on ice. We're doing the actual sequencing here. Yep, okay, good. Looks like all of this is good. Some of these actually stay out at room temperature. Yep, that's good. Okay. How about you? Are you thawed? You're the one that usually takes forever to thaw. Yes, alright. Everything's thawed. Oh, perfect. Yay! Great. I'm excited. Alright, yeah, so if you missed the beginning of the stream, don't worry. If you want to see what I started doing as far as this protocol goes, you can always catch the VOD later on Twitch. I also put it up on my YouTube channel. And that should be going up afterwards as well. There is the link to the YouTube channel. If you also want to join our community outside of the streams, I do have a Discord. If you are interested, you can always join the Discord where you can talk about science and non-science things. It's a nice, chill community, or I'm hoping to have it be a nice, chill community. I just started, so bear with me on that. But yes, I would love for you to join. And if for some reason you guys would like me to post my lovely diagrams, I can do that. This is a Rocketbook, so the QR code, I can actually scan it in and post it up onto Discord if you would like to see it. But yeah, so if you missed what we're doing today we're doing an enrichment and sequencing. So basically I'm looking for specific sequence in the genome that I have using Cas9, which is the protein involved in CRISPR, if you've ever heard that before. And what it does, it looks for it, the sequence, and it's going to cut that DNA where the sequence is, And we're basically going to ligate adapters for sequencing, because the sequencer won't be able to find this DNA unless it has these adapters ligated to it. And it'll go through and sequence basically all the base pairs as part of the DNA here. So if you have any questions, let me know. Let me know. I don't mind at all. And this is a new protocol that we're going through, as in new to the stream. And I'm going to put up our Bert and Ernie. This is our Bert and Ernie double-stranded DNA mascot, so you guys can see it if you hadn't seen Bert and Ernie before. Well, they welcome you as well. The other stream, the past streams that I did cloning technique, which is basically from the 70s and that everyone still uses when we're at the bench. So this is a more modern technique. I keep calling it more modern because sequencing is not as old as cloning. And today we're also doing specifically long read sequencing. I'm going to pull it up. I know I apologize for those who have heard this 3 times now, but I'm just indulging those who are new here. We're doing long read sequencing, and when I mean long read sequencing, we're talking long pieces of DNA. And our lab has done a fragment well over 200,000 base pairs. So 200 kb for short. Versus short read sequencing, which is Illumina, I believe, is about 500 base pairs. They might be over that now. I'm sure they are, like maybe 2.5 kb or something like that. So 2,500 base pairs. And we're doing long read sequencing because we want to be able to capture all the sequence around what I'm looking for the enrichment. So we want to be able to see the genomic landscape, so the DNA sequence around it, because that could be very important. And with short read sequencing, you're going to get shorter fragments where you might not be able to see it. And it's also better for downstream bioinformatic analysis in this case, which I'm not very good at understanding. I don't know a lot about it, so unfortunately I won't be able to answer those questions. But yeah, so we're doing long read sequencing using Oxford Nanopore. That's the technology that we're using in the lab. The other long read sequencing company out there is Pacific Biosciences. That is very expensive. Not a sponsored stream, but this is what we use in the lab in order to do long read sequencing. So if you have any questions about that as well, feel free to ask. But yeah, so let me check and see what we've got on our incubation. So we've got 13 minutes until that is done. And then we'll move on to basically getting a library set up for sequencing. And when I mean library, I mean all the fragments of the DNA that we are creating right now from the Cas9. That's what library means in this case. So we're going to be adding the sequencing adapters, we're going to be cleaning it up and getting it ready to be placed on a flow cell. And I showed this early before, and this is the reason why I wasn't able to stream as early as I wanted to. Like, I wanted to stream this over a week ago, but I had to wait for these to come in. These are the flow cells that we're going to be using for our sequencing today. So we're going to be putting our DNA library that we're going to be making on here. We're going to stick it on our sequencer. I will show you how we actually load all of this on stream, and then it's going to do its thing. It's going to go through and be able to look at all the base pairs of the DNA that we're putting on here. So that's what I was waiting for, these to come in. I was waiting for these to show up. So they finally did, and I'm finally able to do this stream today. I'm so excited. I don't know. What will the count for a partner made? Ah, I see. Welcome. Oh, thank you, Pickle Light Bright. I'm glad you're enjoying it. If you have any questions, please feel free to put them in chat. But yeah, so enjoy. There's other scientists usually hanging out in chat too, and if I don't know the answer, sometimes they will pop in and answer for me. Especially today. It's been very much a Monday for me. Oh yeah, bioinformatics synthetic biostreamer. Oh, so I am not actually a bioinformatician. We have bioinformaticians in our lab that work on this, but yeah, I'm just showing off the actual bench stuff that we're doing in order to do the sequencing. And synthetic biology, I guess you could call my plasmid ligation synthetic biology. I'm making a plasmid, which I'm synthesizing a plasmid. But yeah, so I'm glad you're enjoying it. I'm glad you're enjoying it. Do you have a science background as well? Love it. Yay, yay. So excited. So glad. Here, I'll pop up our mascot. So you can have Bert and Ernie just stare at you. Can be a little unnerving. They can be a little unnerving. I wish they stood. They don't like to stay in one spot. I do. Oh, awesome! Congratulations! Congratulations on getting your master's in bioinformatics. Yay! Oh, that's great. Yay! So glad. Yay! Get that master's done. Get that master's done. Get through grad school. Woof. Yes, you're welcome. I know. I know. Grad school is fun. Fun. Been there, done that. But yeah, molecular biology and genetics. Yes, I'm so glad you're enjoying it. So glad you enjoyed it. I wanted to start this stream because there's a lot of people that have no idea what I do as a scientist at the actual bench, and I figured, you know what? What a better way to tell than to show, right? And talk about it as I go. And I like talking about this stuff. I like teaching. I like teaching. Yeah, exactly. especially when people are like, so are you done yet? Yes, SciComm is so important. I completely agree with you. Completely agree with you. Yes, it's very important. It's very important to get out there and talk about what you're actually doing. Let people see how science is actually being done. I know a lot of people program on Twitch as well. Bioinformatics, I'm not so sure about. I just know there's a lot of computer programming in the science and technology group that I stream in right now. But yes, this is going to be like one of many sequencing streams I'm going to be doing, as this is my main project for our lab right now. Maybe I'll pop in a little bit of cloning here and there. We'll see. We'll see how it goes. It all depends on what I'm working on, but mainly the sequencing. But yeah, I hope you're enjoying it. And please, any questions is great. And I'm so glad that you joined in the chat today. And also to all you lurkers, love lurkers. I'm gonna say it every single time. I love the lurkers. I lurk constantly in other people's streams because I enjoy the atmosphere, and it's so nice to have just a chill. So don't feel like you need to chat or anything like that. Enjoy the stream, enjoy the music, enjoy the vibes, and of course enjoy the science. That's what I'm here for. That's what I'm here for. What is the main project you're working on currently? All I caught was long sequencing. So just a lot. It's basically a lot of long sequencing. I can't go into too many specifics because I don't want to dox myself here in the lab. But we're doing an enrichment of a specific sequence in the lab, and it's we actually have a paper out on this technique, so I wanted to show it as well in case, you know, I can reference it to somebody's like, hey, you can actually watch me as I do this if you have any questions about the protocol. But yeah, so we're looking for a specific sequence within the genome and then doing the long read sequencing to get a better idea of where it's sitting at. So the genomic landscape, as I was saying before, like the sequences around it versus the short read where it'd be difficult to do that. I can give it a little bit more specifics on the sequence. So we're actually looking at it. If anyone's heard of a transposon, we're looking for that, and it's very repetitive sequence, so very difficult to analyze on bioinformatics point of view. Oh, thank you. It actually came out a couple years ago, but thank you. Yes. I'm finally able to stream it. We do this a lot in the lab. This is one of the main things that we're working on in the lab is this enrichment protocol. Yes, so it's been out for a while, but it's always good to get a paper. Always good to get a paper. Where was I? What was I saying? I lost my train of thought. Yeah, so this is a very repetitive sequence, very hard to analyze with short reads because it's repetitive in the bioinformatics pipeline. So that's why long-read sequencing has been amazing for us in order to actually look at the sequence in the genome. It makes it so much easier. So much easier. So let me check and see where we're sitting at. 5 minutes. 5 minutes. And then we can continue on. Then we can get ready for doing our actual library. Yay, library. But yeah, so the next sequencing streams, I'm not sure when it'll be. We're still waiting on some samples for that to come in. We are a human genetics lab, so we work with human DNA in that case. So we're waiting for some human DNA samples to come in order to continue with our sequencing projects. Hopefully those will be in soon. Fingers crossed, fingers crossed. And then I can keep doing it. If not, I definitely have some more more plasmid stuff to work on. We got a positive one from the one I made on stream, which is great. I'm so excited that that worked. But there are 2 other plasmids that I need to be able to create for other experiments that are downstream that I can always add as a stream in case people are interested in doing another plasmid synthesis stream. I'm okay with that. Or a couple of streams. It's like 4 streams just to go through the entire protocol for plasmids. When it comes to sequencing, it's going to be just a day. And then hopefully, we can't do it today, but our lab actually has a channel where we live stream the actual sequencing that's going through the sequencer. But unfortunately, we can't get OBS to work. We had to update the computer and it's not working, so we're still troubleshooting that. So I won't be able to show that today, but I can at least go through it, and hopefully my next sequencing stream will be able to raid that channel, and then you guys can see what nanopore sequencing, long-read sequencing looks like in real time as we're going through it. It's pretty cool. It's pretty cool. But I won't be able to do that. Won't be able to do that today, unfortunately. We'll get that. We'll see. Hopefully we can get that fixed up, and then that'll work next time. So I'm so excited to show like that as well eventually. But we can go through the rest of it, the bench stuff, and then it'll be all on the computer side after that. And I don't do that. I get to hear about it after. I get to hear about it after in the lab when people have finished analyzing the data that I'm generating right here. But yeah, so, oh yeah, which these are all the tubes we're going to be using for the actual sequencing part. I thawed them. They're ready to go as we move on. In that case. So fun. I love the big picture concept. Oh, thank you. Yes. Yes. Sometimes you gotta remember the big picture concept. We get so focused on like the little things, it's hard to remember what the big picture of the project is. But that's what usually you can look through the grants. So we're obviously we're funded by grants in the lab. And usually you can look back at the grants and be like, oh yeah, that's why. That's why we care about this so much. In that case, sorry, I hit my microphone. I didn't mean to. That might have been loud for you all. But yeah, so we gotta be coming up close to being done. Yes, 2 minutes. All right, I'm gonna get the rest of my PPE on, my PPE. We gotta get the gloves on, get that ready, and then we'll be all set for finishing up the library. Or finishing up the library, oh my gosh. Finishing up the Cas9 cuts and moving on to making the library. Alright, so I gotta remember, we do have one more long incubation. This is why the protocol is so long. We have so many long incubations. Then a couple of 10-minute steps. I'm just getting this ready. That's why the stream is going to be longer, just because this protocol takes basically half a day to get through. But I'm so glad you could join me all. Here we go. Let's see. The sample temperature is coming down. I'll grab it out of our PCR machine, and then we'll be ready to go. Going to grab a couple of microcentrifuges because we're going to need these. This one. There we go. I grabbed the right number. That was good. I grabbed the right number. Okay. And I'm going to take the sample out. And right. So, those you just joining, so what we just did was we used Cas9 to basically go through the genome, find the sequence that we're looking for. It should have cut at that sequence, and then we used TAC, so the polymerase, to add an A onto that in order to add the sequencing adapter, because that sequencing adapter has a T hanging over it. So remember, A bonds with T, so we need that in order for the adapter to ligate. So that's what happened in this tiny little tube right here. Now everything else is going to happen in a microcentrifuge tube. So let's move that over. Nope. I'm going to use the big one. I'm just going to bring it all the way up to 200, because this goes all the way up to 200 microliters. And then I'm going to make myself my old very own wide bore tip. Oh, grab it. Using razor blade. Razor blade. Because we don't want to shear our DNA. So using a wide bore tip, meaning that the tip is wider than usual. Let me pull it down. There we go. It will reduce any shear on that DNA, making our very long fragments that we want to keep from breaking. And being careful with it. Alright, so I'm going to add that in there. That was that. Alright, so the next thing we need to add is a specific buffer that has a chemical in it called polyethylene glycol. People have heard of that. I'm sure you've probably heard of it by now. And basically what it is it basically just takes up space in our tube. And the reason for adding this is we want all of our DNA and the adapter we're going to add to be in close proximity to each other, like they get right next to each other, so that they can actually do this bond that we want to make. So if they're right there, it's a lot easier for them to do the bond versus floating off into the oblivion of water. Remember, these are extremely small pieces of molecules that we're working with, like so small. So even like this volume right here, this much volume right here, that's a lot. That's a lot of volume. That's a lot of space. So we want to be able to bring them closer together. So we add this buffer. I'm just pulling it down a little bit. To take up space to allow them to get closer together. We're going to add this next, and it's extremely viscous polyethylene glycol. So I'm going to mix it up with our big pipette, our monster pipette. Because you can't vortex it, it'll make also bubbles. No, we don't want that. But I'm just gonna mix it up a couple times. Because I want to make sure. So, like, see, it's very viscous. Very viscous. It's on the tip as well. So we're gonna add this. Not that much. We're gonna add 50 microliters of this. I can never figure out. There we go. You guys can see it. 50 microliters of this buffer to our DNA mix. I'm just going to call it a DNA mix. Because we have Cas9. That's another thing. You got to be careful when you're pipetting. You got to pipette it slow because it is viscous. And I usually leave it in there to make sure that it actually covers that space. This, see? Tuck to the side. That's how viscous it is. But yeah, you gotta pipette these kinds of liquids slowly. So this glycerol is also a very viscous solution that we use every once in a while in lab. And then we're gonna pipette it directly on there. See how slow, if you could see how slow that was. It's all in there. There we go. So that's the one that's gonna take up space. And then we're going to add our water as well. I need 22.5. Can't really do 0.5, 0.6 is fine. Because I want to bring this up to a total of 200 microliters. Works a lot better. Add that right in there. And now we are going to add the actual sequencing adapter. And this is all from a kit, which I actually need to go this down. I'll be right back. Be right back. Alright, I'm back. Alright, I just spun this down. So 5 microliters of this is our sequencing adapter. They call it ligation adapter. But what it is it's the adapter for sequencing that we're going to ligate onto our DNA. So we're just put this right on top, right here. 5 microliters of that. And then I'm just going to go grab our ligase, so the protein that's going to add the adapter to our DNA is called ligase. And if you were here for the cloning stream, I used that in order to make the plasmid to add a gene to our plasmid backbone. Not that one, this one. We're going to add this. 5 microliters of this. And I add this last because it's a protein and it has to stay cold or it's going to start basically degrading and won't do its job anymore. So I always add that last. And then we mix this way. So remember, don't vortex, don't pipette. We want to not shear our fragments, and vortexing or pipetting will do that. So that's why I mix this way. And is going to continue to mix by putting it on a rotator for half an hour. So that's set. Okay. So this is the last really, I'm going to say really long incubation just because it's a half an hour. So I'm going to put our protein away, put the LNV away, and then we need to make up a solution for the actual flow cell, because we need to actually prime the flow cell, get it ready for sequencing. So I need to make up that solution and we'll have it ready to go. So I don't have to worry about doing that later. So everything should be ready then. Let me put that up. There we go. Yeah. Alright. So I need to look back. This is a new kit, by the way. So I need to look up what the new protocol is for loading, because they have some new solutions in here. Let's see. I'm just looking at. Good. Download the protocol right here. I need to look at it. Make sure still the same amount that we did before. Yes, I believe so. I believe so. Yes, okay. Alright. So, in order to prime the flow cell, I'm going to name this so I don't forget what it is. We need to add 2 solutions together. And this is specific for this Oxford nanopore sequence that we do. It's going to be different for whatever sequencer you're using when it comes to adding stuff to a flow cell. But we have to add this. It used to be called flush buffer. Now they call it, what do they call it now? So with the new kit, they also renamed things. What do they call it now? It is called flow cell flush solution. Flow cell flush. It used to be called flush buffer. So now it's flow cell flush. So we need 1,000 microliters of this, so 1 mil. That's why we're using the big guy. Set at 1,000. And takes up almost the entire thing of it. Good thing we have more of this. 1 flow cell. And it uses almost all of it. So 1,000 microliters of that, and then 30 microliters of the tether, which, to my knowledge, the tether is to help the DNA attached to, they use pores on the flow cell in order to do the actual sequencing. So the DNA is drawn to these pores, and the pore basically grabs it and will sequence the DNA strand. And I believe that's what the flesh tether does, is it helps with that, but I could be wrong. I could very well be wrong. It's all good. Let me just mix that up. So that's ready to go. So this is what we need in order to actually prime the flow cell when we're ready to do the sequencing. I'm going to leave that on ice. And we don't need these anymore, so I'm going to go put these back. Right back. All right. There we go. So that's all set and ready. I have this extra microcentrifuge tube because we're going to be doing, using beads, which I need to mix up right now. And these are magnetic beads. So they look like this. They're like these brown color right here. So these are magnetic beads. They're small beads within the solution. And what it does is it will grab the DNA that we have that should have the adapter ligated onto it now, because that's what we're doing. It's gonna grab it, it's gonna stick the DNA to a bead, and then we're going to do a bead wash. This is called a bead wash. And what's great is that we have, this is our little magnetic stand right here, so these magnets right here. What'll happen is, in the microcentrifuge tube, with the beads are gonna obviously go towards the magnet because they're magnetic, with the DNA attached to it, leaving any solution that it's in, so any of that liquid that's in there is going to be at the bottom of the tube, while the beads of the DNA are stuck here. We take that out, and then we do a couple washes on the beads to get rid of any of the ligase protein that we have in there. We've got the polymerase still in there, we have the Cas9 still in there. The Cas9 is also, we've killed it, essentially, it's dead. Well, not quite. It will be. Just to get rid of any of those proteins, because we don't need that in order to do the sequencing. Just going to get rid of that, get rid of any of the salts from the buffers in there, and we're going to just do a couple washes with this buffer right here. Going to wash that DNA, get rid of all of that. And basically, we're just going to put it in another buffer and get it ready for sequencing. And we don't want extra salts or the wrong salts or those proteins on our DNA when we do the sequencing, because the sequencer will just die. It won't be able to actually sequence. It's gonna get clogged up. We don't want that, so that's why we wash it. The first time I did this protocol, I was mind blown how simple but super effective it is. Yes. Still surprised they don't teach us about this in undergrad lab courses. Sequencing or just the actual DNA extractions. Pickle like right. I mean, this is so easy. Follow the kit, you're good. Honestly, you follow the kit protocol, it's so easy. I know with a lot of new technologies, sometimes people are the magnetic bead protocol. Yeah, so simple. So simple. I mean, you just have the beads and you just little magnet stand, which this is a 3D printed magnet stand that my PI got for us. So you can 3D print these if you don't want to spend a lot of money on a magnetic stand for the lab. They're great. We have a couple of these. But yeah, so yeah, just the concept that academia is a solid 10 years behind industry. Oh yeah, I completely agree with that. I completely agree with that. So this protocol, so Oxford is only a couple years old. Oxford nanopores, so this protocol that I'm doing right here. They're only a couple of years old, and yeah, it seems like it's intimidating when you first look at it. When you're going through, it's like, oh no, it's sequencing. It's got to be horrible, right? It's so easy. It's so easy to do. It's really, you just follow the kit, and I mean, you'll get something. You get something. As far as like setting up a library, so this is a longer protocol just because we're going through the enrichment and doing that. But they have a protocol where it's ready, you're ready to go from your DNA to the sequencer in 10 minutes. 10 minutes. And you're already sequencing something. It's amazing. It's amazing. It is so simple. So easy to do this. So another reason why I wanted to show this too was to be like, look guys, look everyone, it's really not that bad. It is not intimidating at all. I mean, the bioinformatics for me is intimidating just because I am not a bioinformatician. I know a little bit of programming, and when I mean a little bit of programming, I mean a little bit, a little bit. But I don't know anything about how to do the analysis after I do the sequencing. That's all in the hands of our amazing bioinformaticians and grad students in the lab to move forward. But yeah, this is so easy to go through. And the community is also very responsive when it comes to questions and stuff. When people have issues, there's also plenty of protocols that are coming out too for plants, for bacteria, a lot of bacterial protocols. But yeah, so it's not intimidating at all. This is quite straightforward. I think the only hard part is loading the flow cell, and even that is not that difficult. Not that difficult. If you just follow, they have a YouTube video on how to load the flow cell. If you follow that, it's really not that hard. So yeah. When I first started doing this, I was very intimidated, but now going through it's like, oh, so easy. So easy. If you're used to just going through a kit, you're there. You're there. They give you everything that you need, which is great. Even if they don't, the solutions like the beads you can buy like at the big science companies, the Fisher, and there's some money. Shh, save some money. Didn't hear that from me. But yeah, and bead cleanup is used a lot. Like PCR cleanup, although we usually just run a gel because most of the times when we're doing a cleanup, we need a specific fragment and we run it out on the gel to do it. But other labs, you can do a PCR cleanup from this, so short fragments or stuff that you have so PCR is just making lots and lots of copies of a specific sequence using primers. And primers are basically, hey, here's this sequence that I want here, here's this sequence that I want on the other side. The polymerase goes through, the same polymerase that we just used to add the A, and makes tons of copies of that. Tons of copies of that sequence for you. So yeah, so people use this all the time. And this would actually be a very easy protocol in an undergrad lab course. Like you said, pickle light braid. It's been a long time since I've been in an undergrad lab course. For our genetics lab course, we did PCR, so we did polymerase chain reaction. We ran gels. We did a DNA extraction. I'm trying to think, what else did we do? We basically learned a lot of concepts for it, but we did a lot of Drosophila work. So we did a lot of fly work. Fruit flies. If you guys are familiar with fruit flies, the little flies that if you leave bananas out, they all of a sudden show up and they're flying everywhere. We worked with those because they're a big model organism for genetics. Sounds about the same now? Oh, good. So it hasn't actually caught up in the time. Flies and, yeah, extractions. So, yep. It wasn't until, obviously, I got into grad school and started doing my PhD work that, like, oh, wait, there's a lot of other things that happen in the lab. Here, just a second, I need to fix something. Oh, there we go. It fixed itself. It fixed itself. We're all good. Well, I already took my gloves off. That's fine. That's fine. Need a little air. Unfortunately, like, nitrile gloves, my hands are all a little sweaty being in there. Starting to get a little warm in the lab right now. That's all good. That's all good. But yeah, even in, I want to say, I didn't have to take any lab courses in grad school because you're doing research at the bench anyway, so your lab work is all done on your research. But yeah, undergrad was not hugely into big protocols. So much more the current hands-on. Oh, nice in your current college biotech course. Great. Oh, that's awesome. Did you get into any of the sequencing at all, Pickle Light Break? I'm just asking. Oh, a few different. Oh, yeah. NGS. Some theoretical, but surprising amount of actual. Oh, that's great. Oh, that's awesome. NGS, next generation sequencing. So that's when Illumina, that's what I was talking about, short reads, Illumina, is one of the big companies. When they first started doing it, there was Roche 454 pyro sequencing. I'm dating myself right now. It was nice to be able to actually do the sequencing ourselves. Yeah, that's great. That is awesome that you were able to do that. I'm glad to hear that even a small program you were able to do that. Lots of Sanger. Oh, Sanger sequencing. I didn't even touch on that. Oh, goodness. So, yeah, Sanger is one of the first sequencing protocols out there, and Sanger is what we usually do when you're checking a plasmid. When you're doing Sanger sequencing, we're looking at small bits of a plasmid is Sanger, and that one is based on what's the best way of explaining it? Termination sequencing is usually how people will describe it. Just let the caption. Yeah. So with Sanger, it's kind of like when I was describing the TAC where you're doing a PCR, except you use nucleotides that when they're added, they will stop. They will completely stop the polymerase from adding anything else. That's why it's called termination sequencing. And what's special about it is like they're usually called DD nucleotides, so they have an extra D. I forgot what it's called. Dideoxide, I believe. And it's basically the chemical makeup of these nucleotides stops polymerase. It can no longer add anything after that. And so when it does that, it just does this a bunch of times, and it goes through your sequencing. And usually you have to use a primer. So whereas talking about polymerase, so PCR, primers are the sequence that you want to look at. So it's a primer that's up here, primer up here, and it'll go through that sequence. So you pick that spot. It'll just make a bunch of copies of that, and it'll stop at various points within that on Sanger. And the best that you can get as far as Sanger goes is 1,000 base pairs. That's like as much sequence that you can usually get that is reliable. So, but it was one of the very first sequencing techniques out there, and it's still being used today. Sanger sequencing. Like I said, we use it sometimes for doing the plasmids, making sure that that gene is there. You can sequence using Sanger through that, and it'll give you an idea of what you got, if there's any changes that happen, because sometimes you can get like a mismatch on a base pair, and it can cause like that base pair to be deleted, or you can have a base pair inserted. Yeah. Ooh, I'm excited. I always describe it as reading a sentence by shifting the period backwards one word at a time, then stacking the new sentences so you can read the whole sentence based on the last words only. Oh, that's a great analogy. Yes. That is a good analogy. Thank you for sharing. I'll have to remember that the next time I have to describe Sanger. Thank you, Pickle Light, right? I appreciate that. But yeah, it's like, you're right. Moving that period backwards in each of the word. And then you stack them on top, and you can finally have that sentence with all the stacks. Yeah. That's a great way of describing it. But yeah, so the stacking of sentences, yeah. So what's different about essentially this sequencing that we're doing today from Sanger is we don't have the period moving in that case. We're just cutting it, like we're cutting that sentence at the specific word we're looking for. So we're looking for all the sentences that have that specific word, and we're cutting it at that word, and then adding I guess a specific sequencing word at the end of it, so that it'll go through and make sentence stacks like that. So if we're going to use the same analogy, hopefully that kind of makes sense using the same analogy in that case. But yes, I like that. I really like that. An exclamation point. Yes, there you go. Yeah, an exclamation point. Add that exclamation point there. Yes, very, very good. Very good. I love it. I love it. This is great. Ooh, 11 minutes. Okay. I just gotta remember. The timer goes off and we'll be all and ready. But yeah, so, ooh, Sanger. We Sanger, it's kind of expensive if you want to go. So some of the plasmids that we are working with are 10,000 base pairs big, up to 15,000 base pairs big. So Sangering those would actually cost a lot of money for us if we wanted to go through the entire plasmid. We would have to do, it's called primer walking, where you have a primer here, goes through sequences, the primer towards the end, goes through sequences, put another primer, goes through sequences, put a primer here, etc., etc. So primer walking. And each of those times that you add a primer to it costs like $7. And if we're sequencing, I don't know, 5 plasmids that way, it gets very expensive when we use Sanger. So we use Sanger if we have just a couple things that we want to do, but now we've actually have a protocol in the lab where we use this long read sequencing. If we have, I think it's 10 plasmids, we need to sequence 10 plasmids, it actually is more cost effective to do a run on our sequencer, so this long read sequencing, than it is to do Sanger for it. So if I can get 10 plasmids, yeah, it is, yikes, exactly. It's expensive. But if I can get 10 plasmids, then I will obviously do a stream where I show how to do the long read sequencing with our plasmids. I mean, even more is great. We've done 30 plasmids on long read sequencing, and we're able to pick out every single one of those plasmids. It's awesome. And then it costs like negligible. Cost is like so inexpensive at that time. So yeah, but you know, it's got to hit a certain threshold of plasmids in order to make it viable at that point, doing the long read. So you have to pay for the flow cells, you got to pay for the kits for the long read sequencing, and those are, you know, pricey. They're pricey. They're not terrible, but they're pricey. So yeah, you always have that. You gotta always have to do the cost effectiveness when it comes to it. But yay, Sanger! We'll always have Sanger. And then in Illumina, the short read sequencing is even more expensive. Like, Illumina's crazy expensive. The good thing about short read sequencing is, compared to long read sequencing, if you don't care about the long reads and you don't care about the cost, it is more accurate when it comes to doing the sequencing. So it's more accurate compared to long read sequencing. But the accuracy is nice and sometimes, but for what we're doing, we want to be able to see the actual landscape around our sequencer. So long read makes more sense and is less expensive. We can deal with the inaccuracies. That's not a problem because we'll get lots of data. Lots of data. And that will overcome the inaccuracies that that might appear with using long read. We do not incorporate any short read in the protocol. One, because it's way too expensive. And it will take months for us to get this sequencing that we're going to get is going to be done in, at the most, 48 hours. And we'll have all the data that we need. Other labs have done short read. So when this is fair, yeah, fair. Other labs who have done this long read sequencing before when it was first coming out did do QA, like quality control for this kit with short reads. And yeah, there's more inaccuracies. Oh gosh, I can't remember for the flow cell that we're using what the error rate is. For the smaller flow cells, there's two different flow cells that we actually use in the lab. The smallest flow cell has a 10% error rate. I can't remember what the error rate is for the flow cell we're using today, but it's overcome by this sheer amount of data that we're actually going to get. The sheer amount of sequence that we're going to get from the flow cell is going to overcome that error rate very quickly. Very quickly. So yeah, we haven't had to do any short read sequencing to look at our Q&A. Oh gosh, I'm trying to remember. There are public data sets for some of the samples that we used before. So some of the samples we used in our paper that we published on this protocol, there's public data that was based on short read. That's available. We were able to do some Q&A based off of that, but we didn't do the actual sequencing. It's publicly available. And yeah, we were able to get great coverage, great quality out of it, so much data. So yeah, we haven't had to do the short read, but other people have. And we're not planning on doing any short read for these projects. We're good. We also have public data that's available for some of these samples too. So we're all set. We're just going to be doing lots of this. Lots of this. Ooh, 5 minutes. But yeah, so it's very worth it. It's very worth it. It's a very accessible technology too. I know I'm singing the praises of Oxford Nanopore right now. I am not sponsored by them. Not at all. I'm just saying, like, for the stuff that we do in our lab, it is perfect for what we need to do. Now, there are other instances where some labs won't be able to use it. Fair. Very fair. But there's so many instances where you can't use it. If you need a sequencer that is inexpensive, easy to use, and all you want to do is like, hey, I just want to sequence this sample, get the whole genome, done in 10 minutes if you just use the one protocol. And then you'll have the data in 24, 48 hours. Versus, yeah, here if we did Illumina, it would take a month or more before we would get our data. But the short reads and cost a lot more money. But yeah, so sorry, that's my little plug. While I'm here, shameless plug, Pickle Light Bright. If you or anybody else is interested in my past VODs about cloning, they are up on my YouTube channel, so you can go check them out. This is my first sequencing one, so the other ones are basically just going through plasmid creation, transformation, growing them up, because you have to grow them up in E. coli, so we transform them into E. coli, then doing the extraction of the DNA, running them out on a gel after digesting them. And yeah, you're welcome. Yeah, check them out. I know I had someone tell me that, oh, this is so familiar, so nostalgic. When I was going through the plasmid protocol, I was like, yep, a lot of people have done it. A lot of people have done this cloning before, working with plasmids and E. coli. It's a very, very popular protocol that's been around since the 70s. So we use it a lot when we're doing plasmid work, all kinds of stuff. And that's just a separate project in the lab. It is a classic. We should have a list of classic molecular biology techniques. And you've been like, yep, I've done that. But yeah. And I'm trying to think. No, I didn't do that in undergrad. We didn't do any plasmid work in undergrad. We worked with bacteria in my microbiology lab. Yes, I think everyone does that, obviously, in microbiology lab, but not cloning, not the not the plasmid cloning. So yeah, classic plasmids. Oh, okay. Less than 2 minutes. I'm gonna get my gloves back on. Get us ready for moving forward. Now that my hands aren't just warm anymore. Nitrile gloves do not breathe. They do not breathe well. At all. Alright, so the next step once this is done, mixing, is we're going to add a buffer called Tris EDTA buffer, and what it's going to do is it's going to basically destroy the proteins that we have sitting in our little mix, because we don't want those proteins to show up on the sequencer. And the EDTA is just gonna it's gonna basically take those proteins and bind them up, and then they can't do anything, and it's just gonna go out into solution basically. Then we'll add the beads, because we want to get our DNA stuck to these lovely magnetic beads so we can pull it out and get rid of all the protein and that salts from the other buffers. Clean it up. We gotta clean it up. So I'm gonna grab my tea before I forget. It's sitting up above. Tea buffer, here we go. Yeah, this is my collection of buffers. TE buffer. Collection of buffers that I need every once in a while. Handy dandy. And then we'll get that set, and it'll just be a series of 10-minute incubations after this. And then we'll be ready to stick this on the flow cell. And hopefully that'll show up okay. I have to move my setup over to the actual bench where we have the sequencer, so you'll have to bear with me when that happens, because I gotta move the technology around. Alright. That should be all mixed up. We should have adapters ligated at this point, so the adapter should have been added to our, hopefully, enriched sequences, so hopefully the specific sequences we're looking at. And I need to add 200 microliters, so it's 1:1 is the ratio of TE to our mix. All right. Oh. Oh, I don't know if you guys can see it. So this happens sometimes when we have a good amount of DNA. You add the TE buffer, and the DNA actually... I don't know if you can see it. There is, it's right there. The DNA is sitting right there. It's all coiled up right now. And this happens when you add the TE buffer sometimes, which is fine. We just have to do an extra 10 minutes, and it'll go back into solution. I'm trying to see if there's an easier way. It's right there where that bubble is. It's right underneath that bubble. There's the chunk of our DNA right there. So I just need to stick this on the rotator for another 10 minutes. Which is fine. It happens. But yeah, so that's okay. So we'll wait to add the beads, because I want the DNA to go back in. It won't stick to the beads very well when it's a little clump like that. So 10 minutes on the rotator at room temperature will help it go back into solution again. And then we'll add the beads in another 10 minutes. And another 10, then we're gonna be mixing. So we got this 10 minutes. 10 minutes for the beads. For dilution. Okay. Yeah. All right. Yeah, so there's a series of basically 10-minute incubations. So I'm gonna add our next 10 minutes after this, so I know where we're going. But yeah, so this is why the protocol is a long protocol. We do a lot of incubations for it. Incubation. There we go. Oh, gotta love closed captions. Closed caption fails. But yeah, so the rest of these solutions here, sitting here, this is what we use to clean off our DNA. It's called long fragment buffer, once again, from the kit that we're using. The other solutions in here, oh, actually, I don't need that anymore. This is elution buffer, EB. So this is what we're going to put on the beads to get the DNA off of it. And then the rest of these are for when we're ready to actually sequence. So this right here, kind of a milky white substance. These are beads, loading beads. So the beads actually help settle the DNA on the flow cell so it gets to the flow cell where the pores are. So remember, the pores are where it's going to grab those DNA sequences and it's going to pull them through, and that's where it's going to actually see what the base pairs are. And that's based on electric current. Nanopore has a YouTube video about how their sequencer actually works if you search for it. So you can get into the nitty-gritty if you're interested in it. But the loading beads, when we add the loading beads, is basically going to settle that DNA so it gets to the pores a little faster. And then sequencing buffer, this is a buffer that we add that's the right salt concentration, the right salt that it needs for the sequencing to work. And then we usually will use water if we need to dilute or bring up our library to be a certain volume. But in this kit, they said instead of water to use this, which is library solution. So I don't know what's in it. I haven't used it before. This is new to me. I don't know. It might just be more elution buffer. I don't know. But they said to substitute this for water instead. So we're just going to follow the directions and hope they know what they're talking about, which I'm sure they do. All right, I need to put this back. We don't need this anymore. We're actually done with it. This is the adapter. I'm going to go put it back. All right, there we go. So that's done. And if you every once in a while you hear a bang, it's because I'm flipping my microphone up so it'll mute me. So if you hear bang, I apologize. That's just me hitting my microphone on my headset. But yeah, it's nice that the headset that I have, I just flip the microphone up and it mutes it, just in case I need to do a quick one instead of using the mute button on my Stream Deck. So it's real handy. It's real handy. But yeah, okay, so hopefully after the next 5 minutes, the DNA will be back in solution, and then we'll add the beads, and we'll just do another 10 minutes rotating at room temperature to get the beads mixed up. It'll grab the DNA. Then it'll just be washes, and yay, using our little magnetic holder. I love this thing, it's great. It works so well. And with a 3D printer, it makes it very inexpensive. Very nice. But then you'll get to see what magnetic bead cleanup is. So that's always fun. Because you get to see the actual, beads move to the solution. It might be hard. I might be able to do that. So you guys might be able to see it. I'm not sure. We'll see. We'll see how it goes. Hopefully, yes. Hopefully, yes. And then after we clean it up, we'll elute it using the elution buffer. And while it's doing that, we can, I will have to QC the flow cell to make sure that we have a pore. So basically that's just me sticking on the sequencer. And, nope. Sorry, my captions are. Just a second. I'm going to reset the caption. It's getting all wonky. So let's see if it'll fix that. Let me reset my captions. Just hang on for a minute. I'm gonna stop. Just because it's gotten to the end, so it will look weird. Alright, let's restart this. And hopefully it will come back on like it's supposed to and not be all wonky anymore. I've been talking so much. There we go. There we go. Fixed it. Yay, fixed it. Woohoo! Woohoo! Live troubleshooting. Gotta love it. Alright. I feel bad every once in a while this gets, like, off kilter. And it might look strange to you all. But yes. Hope you all are enjoying the music and the vibes in the channel. Once again, I'm glad that you could join me today for this long protocol. It's been nice to be able to finally do this stream. I've been looking forward to it. And then hopefully many more. Many more. Many more. But I'm actually going to grab a quick drink. And I will be right back if you guys would like to get a drink yourself. I'll be like 2 minutes. 2 minutes, I'll be right back. Alright, not even 2 minutes. It was quick. It was a quick drink. I was feeling a little parched. But yes, we've got minute 50 seconds. Get my gloves back on. The DNA should be back in solution now. We'll double check. We'll show you when it comes off the rotator. And we can get to cleaning it off. So basically right now, we have DNA with the sequencers added to it. We have a library. A sequence adapters ligated to it. So we have a library. We have our DNA with the sequence adapters attached to it. That's a library. It's just a bunch of fragments ready to be cleaned up and stuck on the sequencer. We're just cleaning it up now. Pretty simple, right? Pretty simple. It's very easy. Very easy. So you don't have to be intimidated by it if someone says like, oh, sequencing library. Like, nah. It's simple. Simple. You can do it. You just follow through with the kit. It's all good. It's all good. Then we'll be all set. Oh, the one thing that, yes, I definitely want to check the concentration. So, concentration, we're looking at concentration that basically says how much DNA we are going to have in the tube when we're done. Because we have to load a certain amount of DNA onto the flow cell, and my guess is we're not going to be able to add all of this library, because what's nice about these flow cells is you don't have to put a lot of DNA on it in order to get a lot of data out. So that's nice. So I definitely need to check. Based on our previous concentrations of libraries, pretty sure I'm not going to be loading this entire thing on it. We usually get micrograms, so micrograms of DNA. That's a lot of DNA for a flow cell. All right, let's grab it. Let's grab it. Yes, yes, yes. So that little white, it kind of looks like a white fuzzy, is gone now. So that means our DNA has gone back into solution. Great. So let's add the beads. 120 microliters of beads. I'm just gonna just make sure the beads are in there, because they will settle. There is a weight to these beads. Yeah, you see. Look, not a colorless liquid. Not a colorless liquid. We know something's there. I'm just gonna add it directly to it. There. So you just mix it up. So this is going to go on rotator for 10 minutes. It's going to grab our DNA onto these beads in that 10 minutes. And we'll be able to pull it off and clean it up. So I'm all done with this. I'm going to go put this back. I'll be right back. Alright, so we're all done with the beads. All we have left, so we started off with what seemed like a lot of tubes of stuff. We're back to just a couple. We basically used most of everything that we're going to need for the sequencing. The rest of this stuff is just basically for getting it on a sequence, the flow cell. And then we'll be all set. But yes. I know, it's gone over 2.5 hours. 2.5 hours. I scheduled this for 4.5 hours. I wasn't sure how long it was going to be while I'm recording. I know it's about 3 hours when it's just me at the bench doing it, so I gave myself a little bit of leeway in case they wanted to do some extra explaining of the protocol at the end, and for obviously questions. If people have questions as well. So it might not be that long. It might be that long. We'll see. We'll see how it goes. So yeah, if you have any questions, feel free to pop them in the chat. Not a problem. Not a problem to go through them. Since we're all doing science here. Science at the bench. We love science. There you go. Wheel of Science. Oh, closed captions. The things you think I say. Sorry, I took off my glove to check my computer. I'm trying not to contaminate my computer with science things. That's why I took it off. And that's why we go through gloves. Like this whole thing of gloves. I will go through that pretty quickly. Because you're not supposed to reuse gloves. It's a big no. Don't reuse gloves. These are meant for one-time use. They're disposable. You don't want to reuse it just in case you got into some nasty stuff. Don't do that. Don't do that. Don't do that. Follow our lovely, I mean, it used to be OSHA, but now it's EHS, Environmental Health and Safety. But yeah, I hope you guys are having a good day today. Night, morning, whatever it is for you. I think I'm doing a lot better than I did at the beginning of the stream. For those of you, if you didn't know, at the beginning of the stream, I talked for like 10 minutes without realizing my microphone was off. So, it started out as a good morning. Definitely a Monday. I'm feeling a lot better now. Getting into the groove once again. But sometimes I feel like, or you always get a little anxious before you hit the go live button. At least for me, when it comes to streaming, which it should be. I'm excited about doing it. But it's just like public speaking. Like I like doing it, but I'm always anxious right before I do it. Just because, you know, it just happens. But once you get into it, that anxiety goes away, at least for me, and I start feeling better, feeling more calm. I just remember talking at conferences right before like getting up on the stage. You're just like, my hands are all sweaty, my heart's racing, and then I finally start getting into my talk, and then I feel like, wait, I'm done? Out of body experience kind of thing happening. I started talking and then I don't even remember what I was saying. And then the questions start coming. I'm like, oh yeah, I'm here on stage actually talking about this. Okay, yeah, now I know where I am. I don't know if anybody else feels the same way. Even though it's fun. That's just how it is. But yeah, I feel like I'm doing a lot better. No, at least I'm seeing that my microphone's going, so that's good. I'm not talking to the void. That was this morning for 10 minutes. Oh, I still can't believe that. But it's all good. Not a problem. Everything can't be 100% awesome. But it's fine. We recovered. And you know what I forgot to do? I forgot to turn on my timer. Oh gosh. How long do you think it's been? About 5 minutes. As long as it's been at least 5 minutes. There we go. I said I was doing good. People. Oh, thank you, Pickle Light Bright. Thank you. I just forgot to turn on my timer. It's all good. Oh, it happens all the time. It happens all the time. And that's when I'm usually like, oh, why'd you do that? How long has it been? Looking at my watch, trying to figure out, oh, it's been long enough, right? It's been fine. We've been fine. As long as it's 5 minutes. As long as it's 5 minutes on there, that's good. But I try to do 10 minutes just to give it enough time. But if we just did it for 5 minutes, it'll still be okay. It'll still be okay. I just like to give myself that extra time just in case. Just in case. But thank you, Pickle Life Rick. Thank you. But yeah, you get to see all my bumbling today. It happens. It happens. I do the same thing when I play video games, so it's all good. I bumble through video games a lot. Maybe that's why I decided to do this instead of video games. Oh. Whoops. Excuse me. I just ran into it. We have a trash can right here next to my bench, so I ran into it. That was that noise. If you heard that, I didn't mean to. But yeah, so let's see. I'm excited to show you guys I hope you can see it. I couldn't actually test it earlier on if you would be able to see me loading the flow cell, because I would have to load the flow cell. But hopefully you guys will be able to see it. I might be able to finagle how I hold the pipette and stuff for you to see it. We'll see. We'll see how it goes. Just the way that the camera's set up. It might be difficult, but I'll try. I will try my best. And we might be able to figure out something else for next streams where I'm doing the actual flow cell loading to help with that. And then might be able to show using the other flow cell, which is cool. So we're using bigger flow cell. I'm gonna show it again. I'll show it again. This is the flow cell that we're using. This right here. We're gonna be loading this bad boy right here. This is called a min ion flow cell. But it also uses a smaller one called a flongle. A flongle. Not fungal. Flongle. Oh no. Flongle. Flongle. There we go. Oops. Oh, I'm sorry about that. I'm not sure what happened there. I'm sorry about that, Pickle Light Bright. I have Streamlabs set to certain moderator things. I can see if I can fix that. I didn't mean to time you out. I apologize. Hopefully, okay. I'll have to fix that. I'll have to fix that off stream. I'm not entirely sure how to do that right now. I'm sorry, Pickle Light Bright. I didn't mean to moderate you like that. Let's see if I can. Ugh. I don't have the time right now. But I will keep that. I will keep that in mind. That's a note to me to fix that. No, it's fine. I mean, it's okay to write in all caps, but I didn't realize that it was going to be that strict about it. So I will fix that. I will fix that. So people can write in caps if they wanted to. If they wanted to. Streamlabs bot. You're mean. You're mean, Streamlabs bot. So yeah, whatever your message was, just if you want to put it back in there, you could do it without caps if you wanted me to see it. And then I'll fix that. I'll fix the moderator settings on that for next time, so you don't get yelled at by Streamlabs bot. I mean, it's Streamlabs. I think it's Cloudbot, but it calls itself Streamlabs. It's fine because I use Streamlabs for streaming. That's why. So I will fix that. Ooh, okay. Ready? We're gonna take it off. 2, 1. All right. Time for the bead cleanup. All right, so this is what it looks like right now. So all the beads are thoroughly mixed in there, and they should have grabbed our DNA, which is good. So we just stick it on here. Maybe you guys can see it as the, maybe. It's gonna be kind of hard to see. It might be easier this way. It might actually be easier this way. All the beads are gonna head right to the magnet, and you'll be able to see them as they congregate right there, towards the bottom of the tube. It takes a little bit. It takes like a minute or so for them to all get there, and then all that's left, other than you'll see the beads, is going to be the solution, and in that solution is going to be the proteins, the Cas9, the ligase, and the Taq polymerase, which have been bound up by that EDTA and can't do anything. And then the salts in the buffer, all the buffer that we're using, the water that we put in there. Oh, it's almost there. Just a little bit longer. Hopefully you guys can see it. It like went from kind of a cloudy brown to now it's getting very like darker brown. That's all the beads going towards the magnet. A little bit longer, it's almost there. I can see it if I'm looking this way, like how much of the beads are actually still in solution right now. So close. So it's about a minute or so. So close. Here we go. Okay. So, and they're going to stay there. They're going to stay there. What I'm going to do is we're going to put this back up to 200. I'm going to, oh, I forgot to get a waste speaker. Got to get rid of the solution and put it somewhere. So I use a waste speaker for that. Waste speaker. There we go. I'm just going to pipette all that out. So I usually hold it this way so I can see. Maybe you guys can. There we go. So I don't touch the beads. It also gets rid of the polyethylene glycol that we added earlier too, because that will definitely kill our sequencer. Well, not the sequencer, but the flow cell, because that is the one that takes up space, right? We don't need it taking up space on the flow cell. We just want the DNA and the solution that it requires on there. So there. So now the solution's gone. All that's left are the beads. So we're going to take it off. I'm going to add our wash buffer. 200 microliters of it. We're going to do this 3 times. 3 times. I just pipette it onto it. Mix it up. Do a little mix. A little tap. Right back on. Right back on there. Now, though there's some stuff on the top, which is fine. What I'll do is I'm going on the last wash, I'll spin it down, make sure I get anything off the top. And we got to get rid of all of this buffer right here because this is a wash buffer. So we don't want the salt from this, we don't want carried over into our actual sequencing buffer because it's the wrong concentration of salt. And it won't sequence well. So we want to make sure we get all of that off. So I'll spin that down and do one last bead. Clean up with that. So he's going a little faster this time because we don't have the polyethylene glycol on there. The polyethylene glycol makes it viscous, so it takes a little bit longer for those beads to go through. There we go. That was much shorter. It's just the same exact thing. I didn't even change the tip because we're just going to be taking it right out. I'll change the tip now because it's considered dirty. Because we're washing it. And I do 3 washes because I want to make sure it is all that polyethylene glycol is gone. Because it's fine. I didn't need to get rid of the tip, but I did anyway. Because it really will. It'll kill those pores on that flow cell if it's on there. So I do 3 washes just to make sure. Yeah, and after this, we'll do the elution. And then I'll get the setup ready for loading the flow cell. They'll have to give me like about 5 minutes in order to move everything over there and get it all set up. So I gotta move the camera. I gotta move my laptop over there and get that all set up and make sure that the camera's being picked up because I have to use a different USB port while I'm over there. And sometimes Streamlabs is like, what? You want to do what now? Alright. But yeah, these bead cleanups, so easy. So easy. Alright, the last one. And then these should be all cleaned up after this. Alright, I'm gonna let that sit. I'm gonna go put the wickets for all done with it. And then we'll be all set to, I'll do it another spin. We can loot it off. And then hopefully we'll have a wonderful concentrated library at the end. Look at that, look at that. And so well. So I'll spin this down after I take this off, make sure I get everything off the top, put it on the beads one more time, get rid of anything that could be left over. And then we'll do the elution buffer, which is this right here. And that will remove the DNA off of the beads. And a little bit of heat as well. Because I'll put it at 37 degrees Celsius for 10 minutes. And that helps elute the DNA off of the beads. Right. Okay. Okay, let me go spin this down, and I will be right back. All right. So there is a little bit left. You can tell that there's a little bit of solution left. So I just do this. I hold it because if I just did that, sometimes it doesn't get the beads off. So I want it actually a little bit closer. So I need to get just that tiny little bit. I'll use more. I'll put that for that. And sometimes I move it because I want to get it out of that solution as much as possible. Because sometimes, even though I have it not even touching the beads, it'll still like to be pulled into that solution. So I try to move the tube up a little higher and higher once I have most of it out of the solution. Just to make sure I reduce the chances of the beads being sucked up. Because we still have the DNA on that bead, I don't want to lose it. There we go. Alright. That's all gone. That's all of that solution gone. Okay, so now we're gonna add solution buffer. Oh, how much solution buffer? Just a second, I need to check. I forgot to check that. How much solution buffer do I need? I mean, checking in my protocol here. 15 microliters. Okay. 15 microliters. There's a solution buffer. I'm gonna mix it up just a little bit. Okay. And then I'm gonna stick it on the beads as much as possible. I'm gonna tap it down. And then I need to, so I'm trying to be careful with it. This is why I'm kind of mixing it this way. Because I don't want to be too horribly rough. I still want to keep those long fragments. We got a little bit of stuff the side. If I can get it off the side. I think I'll spin it down real quick too. But then I'm going to, so there we go. So the beads are basically in that elution buffer. Whoop. So now I'm going to go spin it down just to get, we got a little bit on the top. I want it all to be down here so it'll be able to elute better. I'm going to spin it down just real quick to get 37, and that'll incubate for 10 minutes at 37 degrees Celsius. I will be right back. All right, setting my timer. Setting my timer so I don't forget. Okay, so now that's incubating. So for 10 minutes at 37 will really help get long fragments of DNA off of the beads, which is what we're trying to do right now. And this is just something that we've seen in our lab. If I did it just at room temperature, sometimes you'll see the beads are going to be like all balled up, meaning that there's still DNA attached to it. So those, and because it's long DNA, it's like stringy, is the best way to describe it. And we want as much of that to come off the beads as possible in order to get it on the sequencer. So doing the 37 for 10 minutes really helps, And it might cause a little bit of fragmentation, but not a whole lot where it's going to destroy every single one of those long fragments, but it'll help it get off of those beads, which is good. I'm going to put the elution buffer back since we're done with this. And then I will do, so basically it's a 20-minute total for the elution, 10 minutes at 37 and 10 minutes at room temperature. Just something that we've done in our lab. It aids in it by having that extra 10 minutes in the elution buffer. So 20 minutes total, we usually get really nice results from that. Now, while we're waiting for that, I'm going to get, so putting this at 199. We're going to be, I want to check the concentration of the library once it's done. And that means we're going to use our qubit. And if you missed it, we use the qubit in, I think, my third stream to check what the concentration of our plasmid DNA was after we extracted it. So basically what it is the machine that checks nucleic acid concentration. So how much of a nucleic acid we have. And it uses specific dye, specific buffer. And the one I'm doing is for double-stranded DNA, because that's what our library is double-stranded DNA. So it's 199 microliters of the buffer and 1 microliter of this dye. And this dye is specific to double-stranded DNA. You can get one for RNA, you can get one for single-stranded DNA. So that's what's nice about using the qubit, is it's specific to the exact nucleic acid molecule you are looking at. And that's why we use it to check our concentration. Let me just add a microliter of that. And just mix it up. And then it's ready to go. And I'm just going to put it off to the side for right now. And then I'm going to put it back. So we'll need that. So what's nice is we only need to take a microliter of our library and we'll add it to the tube. 2 minutes to allow the dye to incorporate into the DNA. And then I'll check it on our machine and it'll tell us how much DNA we were able to get off of our beads as far as the library goes. And then from there, figure out how much we're actually going to stick on the flow cell. Because we won't be able to do the whole library. It's just not going to work. But yeah, all right. So that's what we'll do after the elution's done. But okay, so hopefully, goodness, I want to try to figure out what's going on. Hopefully Pickle Library hasn't completely modded you out. I hope not. I don't have my full setup on right now, so I can't really check it easily. Let's hang on with me, everyone, while I look into okay. Good. Like I said, I was hoping that it didn't mod you up. That's fine. Troubleshooting your code, have fun. Yay! Troubleshooting code. The small amount of programming. I remember that. Usually I put a semicolon where it shouldn't have been. That's usually how my code ended up dying on me. So glad you're still here. While I figure out how to do this stuff. I think I have to do it on the actual dashboard. So I'll have to do it. It's fine. I'll do it later. I will do it later. Write myself a note while we've got some time right now. Ooh. I've got... here we go. Let's see. Writing myself a note. Fix cat mod settings. There we go. And pin it. So I will do that right after the stream while I'm waiting for this VOD to upload to YouTube, because it's going to take a while. It'll take a while, but it will be up later. For those of you who are new, hopping in to listen, if you would like to catch up on this stream or my past streams, I have a YouTube channel where I put all my VODs for Twitch on there. My previous ones were doing cloning with the plasmid at the lab, and that took about four streams to go through the entire protocol. And then this one will go up, and all my next ones will obviously go up. But yeah, so if you missed the beginning of the stream, it also will be up on Twitch as well. Sorry, I was checking something. I got distracted. Excuse me. Squirrel. Squirrel. So many things. So many things happening. That's all good. Oh gosh. Okay. Oh goodness. Things to catch up on. Like I said, this is a long stream. So if you cut out, it's all good. I understand. I get it. It happens. We've been streaming for 3 hours now? Well, I know I've been recording for 3 hours now. Not including the beginning, 10 minutes of me just talking to nobody because I muted my mic. Haha. All good. So yeah, so this is basically gonna be about 4 hours. 4 hour stream, I'm guessing. Unless we have a good discussion about the actual protocol afterwards, so not a problem. Like I said, I scheduled this to be about 4.5 hours. I wasn't sure how long it would actually take to go through this, so it's all good. It's all good. We're almost done with our first incubation. And then I'll go grab it, incubate it for another 10 minutes. Yep, already got that set. And then after that, we will check what our concentration is. And then I will set up for doing the actual loading on the flow cell. But yes, floor self. Oh, closed captions. Closed captions, you're the greatest. You give me so many laughs to myself. And hopefully others as well. The closed caption fails. Especially the science. Science terms, does not like it. Does not like. Yeah. All right. All right. Less than a minute, and then I'll go grab it. Oh, I'm so excited. I'm so excited to try to figure out the loading with you guys. Hopefully you'll be able to see it. And then hopefully we get some really good data out of this too. I'm sure we will. I'm sure we will. It's a new kit, that's why. That's why. I'm a little apprehensive, but we're gonna be good. It's gonna be good. We've always had decent results from our sequences, so. Almost. I'm gonna just walk over. There's gonna take me about 10 seconds to get there, so it's all the way at the other end of the lab. All right, I'm gonna go grab that. I'll be right back. All right, I'm back. So, first incubation, now we're going to do another 10 minutes at room temperature. As you see, a little condensation at the end. So I will spin this down again before we put it on the magnet one last time. So, to elute it off the beads, what we're going to do is we need to get the DNA that is hopefully gotten off the beads, should be in solution. So we just need to pull the beads away and then take out the solution and put it in our new microcentrifuge tube right here. So hopefully, it worked well, and we'll be able to get it off. I might actually use this just because I can make a better wide bore tip with this one. And we can use these tips. Just works a lot better than the P20 tips sometimes. Because these are, I'm going to sacrifice one to show you. See, these are a lot shorter, a lot smaller, so I have to actually cut a lot higher up, and I won't be grabbing all the solution that I want to by cutting that high up. But this one I can do about the same pore size, the same space size, but further down. So I think that's why I'll do that. Makes it a lot easier. But yes, I'm so glad you guys are here today. And for all of you that have stuck around for the entire almost 3.5 hours, thank you. Thank you. I'm very happy that you were here to join. And got to listen to me go through this protocol. From basically beginning to end. If you plan on sticking around for the whole thing, that's amazing. You are amazing. Thank you so much. I appreciate it. I feel loved that you enjoy listening to me talking about science. And watching, of course. And also enjoying the music. I mean, if you're here for the music too, that's okay. I'm okay with that. I'm okay with that. Can like get a little bit of science, little science mixed in with the music. A little bit of like science osmosis. I'm just going through, I'm checking over my notes one last time to see if there's anything completely different to loading than compared to before. That's what I'm doing right now. So that's protocol. That's what I'm looking over right now. Just to see if it's any different from how we did it before. It doesn't look like it. It looks like it's the same. Yeah, okay. That's good. That's good. So I've loaded these before, but you never know with the new kit. And these are also, I guess, an upgraded flow cell compared to what we used before. You always want to double check and make sure that there isn't a surprise. Like, oh wait, you need to do this now. Compared to how you loaded it before. So that's why I'm double checking with the protocol that I downloaded and printed out. Make sure I'm doing it correctly. Because, you know, bumbling through isn't always that fun. But that's okay. That's alright. We'll make it through. But yeah, so. All good. We got about 6 minutes. Does anybody have any questions? Like anyone new popped in and is like, what is going on? I have no idea what's happening right now. Or any questions about what I've talked about before. Feel free to ask. Feel free to ask. It is all good. And then hopefully my transfer of technology over to where the the flow cell, the sequencers are will go okay. If it doesn't, I will probably post in Twitter, which I'll put up my handle here in case you want to follow me on Twitter. If it doesn't go well and something crashes, I will post an update on my Twitter. Just so you know. You know, like, oh, she vanished off the face of the earth. Why? That would be why. Or at least the Twitch universe. That would be why. So it can be all set for that. I'm just warning you, I don't post a lot. It's usually just when I go live. I will post on Twitter. I do a lot of likes. Likes of other people's posts. But if like something comes up, because Twitch doesn't really let you send out post notifications to your followers, I will post it on Twitter in that case. Or if you wanted to join the Discord, I do have a Discord community. If you would like to join outside of the stream, feel free. I will post messages in there too if something goes wrong. That's not a problem. It also posts that notifies you when I go live as well, if you don't want to do Twitter. But yeah, if you're enjoying the stream and you haven't already followed and would like to follow, go ahead and hit that follow button if you want to get notified the next time I'm live. Oh, sure, Pickle Light, Bright. I'm glad you could be here. And I'm glad you enjoyed the stream. Thank you so much. Hopefully I'll catch you again, but if not, then that's not a problem. I'm glad you were here today. If you'd like to see the end of the stream, it'll be up on my YouTube page later, or you can see it on Twitch as well. But thanks for being here. Appreciate it. Enjoy the rest of your day, evening, morning, wherever you are. But yeah, as I was saying, if you would like to be notified the next time I go live on Twitch, you can hit that little bell, and it'll let you know when I go live next. And I'm not sure about this week. I'll have to see. I still have to go through my plans for the week. I might do another plasmid stream later in the week. Unless we get the samples I'm looking for, or I'm waiting for, to do more sequencing, and then I'll do another sequencing stream. But I can always do another plasmid stream. Like I said, I've got 2 plasmids. I got the one that we were working on. Yay, we got one that looks good. So excited. But I've got 2 other ones that I can work on that are the one that we weren't able to get any colonies for after doing the transformation. So I have to redo that ligation, so redo that design. And then another one that has been in my backlog that I can add as well, that also just needs a little tweak to how I design it. So I can always do that this week as well, if that sounds good. We can do another one of those. We can bring out our other mascot, Eli E. coli, if people would like to see Eli E. coli. We've seen Bert and Ernie today, since we're doing DNA. But I have not added Eli E. coli, because we're not working with E. coli today. Just DNA. So I can always do that. Not a problem. Ooh, 2 minutes. Alright. Alright, I'm gonna get my gloves on and get ready to go and see how well DNA is gonna loot off these beats. Hopefully it'll go very nicely. Sometimes I tell ya, it will stick to the beads, and sometimes it does not want to come off, which means that there's long fragments. But let me do my wide My homemade wide bore tip. Homemade wide bore tip. Works great. Works great. Let me get that all set. There we go. So, yeah. I'll do a quick little spin down right before, just in case. And then we'll put it back on the magnet, and we'll take out the solution, which should be our DNA. Stick it in the new microcentrifuge tube. And then I'll get ready for loading the flow cell. That'll be awesome. That'll be great. I'm excited. And we'll have to mess around, move technology around, but that's okay. That's okay. Okay. Hopefully it works. Hopefully I can show you how to load it. And then it won't be so intimidating if you end up using Oxford Nanopore in your lab, I guess? For other lab people? If you're ever interested in it, you're like, oh, that sounds like something we might be able to use in our lab. Look into it. Look into it. It's brought to our lab wonderful, wonderful results. Alright, alright. So close. 2, 1. Done. Alright. Let me go spin this down real quick, and I will be back. Alright. Okay, let's see how nicely it's going to loop up the bead. So I'm going to do the same thing that I did before with the last wash. Where I'm going to hold it against the magnet. So hopefully the beads will collect right there. Looks like they're doing good. I don't see any clumps not wanting to go to the magnet, which sometimes happens. Well, sometimes happens. Alright, now I'm going to start moving it up slowly to help it get away from the solution. So it'll make it a lot easier to pipette it. And hopefully not grab any beads, because once again, at this stage, sometimes, even if you're careful, you can sometimes get beads that don't want to move. Alright, let's try it. Let's give it a go. Go real slow. Okay. So I got a little bit right there at the bottom. I can see it. See if I can grab it. I can grab that too. This is where you just gotta be careful. I don't want to get any beads, because the beads will also not be good for the pores on the flow cell, because it'll take up space that the DNA is supposed to be at. Ah, nope. Okay. If I put it back real carefully. Let it sit there. Can get anything else off. What? Nope, there's just a little tiny bit off. Okay. All right. Okay, I'm gonna keep that just in case. Tap this down, tap, 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 tap. Tap, 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 tap. All right, so I'm going to check concentration of our library, which we take a microliter of our library, which is a clear solution, as you can see, clear solution. And add it to the cubic tube that I made up, where it has the dye, and mix it up, and we let it sit for 2 minutes so that the dye can incorporate into the DNA. Then I'll go use our machine and be able to check it and see how much we've actually got in our library here. Let's see. Just checking to see if we've got anything else just sitting here. Sometimes. Like I said, it doesn't like to come off the bees, but it actually looks pretty good. So, yay! Hopefully, we'll get a good concentration and figure out how much to load once we know what the concentration is. So, with these flow cells, like I said, these are updated flow cells. No, grab that because I need to bring the flow cell over there. They require even less DNA than the flow cells that we were using before. So I can't load 250 nanograms. 250 nanograms is the max to load onto these flow cells. So I can't load more than that, or it'll not be as efficient at sequencing, because it'll just be too much DNA and then it'll just overwhelm the pores. So we gotta check and see. That's why I need to check the concentration, because if I just loaded all of this and it is crazy concentrated, it won't be a very good sequencing run, because it'll just overwhelm the pores and it'll sequence horribly. We've done that before. We have overloaded a flow cell before, and we saw that. You can see it when it's sequencing that you've done it. You've been like, oh, that's terrible. All right, so a couple seconds here. And then I'll go check it and we'll figure out how much we're putting on our flow cell. All right, give me a minute and I'll be right back. All right. We got 62 nanograms per microliter. I'm gonna write that on the side of here just so I remember. And then this is what's today's the 27th? Date on there, so I know which one this is. All right, so let me do a little bit of quick math. How much do we have in here? So we want to load 4 microliters. That will be 250 nanograms. That's how much we want to load from our library that we just got. Now, I have more than 4 microliters, so I'm going to make up a separate tube of our library that includes the sequencing buffer and the library solution, which is taking the place of water in this loading. So I'm going to make that up. Let me just do quick calculation, find out how much we need of that. Oop, put away my calculator. What's it called? Okay. I'm just writing this down in my lab notebook so I do not forget how much we use. All right. So, to make this up, we need our 4 microliters of the library. Okay. In here. And we also need 8 microliters of this library solution, which would take the place of water, because that's what they want us to do with this kit. Not sure what's in it, to be honest. Not sure, but they said to use this instead of water, so we are going to follow the directions. Let's add that. And then we want to add the actual buffer solution for sequencing, which is a sequencing buffer. And that is 37.5. Can't really do 0.5, so do 0.6. And we're going to add that. There we go. So this buffer is the correct salt and concentration that it needs to be. And I'm just going to do a quick mix like that. All right, so the rest of this is going to be done over at the actual sequencer. So give me a couple minutes. I need to move everything over there and get it all set. But hopefully I'll be right back, like 5 or so minutes, and we can get this loaded up. So I'll see you in a bit. All right, let's see. Yep, sorry. Let me change it up here so it's not seeing my camera. I had a feeling I was going to do that. Oh, goodness. Okay. Just a second, everyone. Doesn't want to see my camera. Let me plug it in again. I had this all figured out earlier. I tested it earlier. Oh, goodness. It doesn't want to find my camera. Oh, I'm sorry. I am sorry, everyone. I think I need to restart Streamlabs in order for it to see the camera, unfortunately. Let's see if I can do something else here. Let's see, before I restart Streamlabs, which means I'd have to end stream and then restart again. Yeah, okay. So my other app is showing it, which means it's not showing up in Streamlabs. Hello, is everyone still there? Sorry about that, my laptop decided to die. Died. I forgot. It needs to be... It needs to be on power. There we go. Now I just need to reset. Just a second, just need to reset web capture. Oh. Nope, nope. Select up. Am I still here? Yes, okay, okay. Sorry about that. Hopefully you can hear me. I mean, it shows that my microphone's going. Let me fix my captioning just a second. Hopefully it is back up and running. Yes, sorry about that. My laptop does not run off of... It runs off of battery. I forgot to bring over the power source when I moved this over. So it kind of died on me. I apologize. We're fine now. And you can see our sequencer. So we're all good. Alright, so yes, this is the sequencer that we're using. So this is called a min-ion sequencer. And hopefully I'll be able to show you how we're loading it. Right now, the flow cell is QCing, so we're checking how many pores are in the flow cell at the moment. And while we're doing that, I'm going to get our pipettes ready. So I need to set this 200. I have this zoomed in a little bit, so hopefully it still focuses nicely. And if for some reason I am stuck, maybe refresh the stream just in case, because my laptop died, so that could be why. So we just gotta wait until it finishes checking our flow cell here. So it's just going through using the software, checking to see how many pores we have that we can use for sequencing. So remember, we're using a pore, and the pore will grab the DNA and run the DNA strand through it. And based on the changes in electrical current, that's how it decides what the base is. If you need more information about the nitty-gritties on how Oxford Nanopore does the sequencing, they have a YouTube video about that if you search for it. But yeah, so we just need to wait for this to finish, and then we can do the load. And basically all it does, we have to first prime the flow cell. So we have to basically put solutions through it, get it ready for sequencing. And that's the very first thing we have to do. Then we can load the library on. And I can't remember. We'll add the beads that I was talking about right before we load it, because the beads are like the magnetic beads where they will settle. And we don't want them to do that before it starts sequencing. So let's just give it some time to finish doing what it does best. Hopefully we'll have a good number of pores to sequence with. So when we first get these flow cells, I actually go through, and you need to QC them, so check how many pores you get when you actually receive them from shipping, because they are warranted for a certain number, meaning that if it's below a certain number of pores, when you QC it, you can get a replacement. But you have to do it within a certain time frame after you get them. So I've initially QC'd this at 1,607 pores. We will see what it QCs up as today. And pore number changes. Sometimes we gain pores, sometimes we lose pores. I just don't want to see us lose 1,000 pores. That would be bad. That would be bad. Because then I'd worry about this flow cell and I'd have to grab another one and check that one too. But it takes about 5 minutes. I've already got this started, so it shouldn't be much longer to go through and QC the flow cell in this case. So hopefully it'll be good. Really good. But thankfully, I have to say that my laptop getting knocked out, at least Streamlabs found my web camera. So I wouldn't have to exit out of Streamlabs. But let's see. Still doing his thing. Because we have all of this is attached to its own computer, where it captures all of our sequencing information, all the data that we get, and moves it off for the downstream bioinformatic analysis. So yeah, I'm excited. We'll see how it goes. Maybe this is the last one. So it basically goes through a series of scans on the flow cell. And it, I think it runs a current through them. I'm not entirely sure. To check to see which ones are available that are still working on the flow cell, like which pores are still working. And then it will spit out a number once it's done of how many are available to do the sequencing we have. Come on, you can do it. You can do it. Give us a good number. Give us a good number. Yay, okay. Oh look, we gained 2 pores. 1,609 pores. All right. I'm excited. Okay, so now I'm going to show you how to prime the flow cell. So this is what it looks like while it's in the sequencer. So this is our lovely flow cell here. And it's kind of upside down, but it's all right. So what we want to do first is we move this. This is a little slide thing. This is a waste port right here. And there's usually a bubble that is sitting in there that they tell us to get rid of. So I set this at 200 microliters. There you go. And we're going to put this right down into the waste port. So we have the pipette right down the waste port. And what I'm going to do is I'm going to try to suck up that bubble by slowly increasing the... Oh, I saw it. Want to slowly increase. There we go. So that should have gotten rid of that bubble. See? So this is the yellow solution in there. I wanted to see that, meaning that any kind of air that could have been in there has already been sucked up. So I slowly increased the volume on the pipette to get that air bubble out. All right, so that's done. Now we're going to add that flush buffer. So we need 800 microliters. There we go. So we made up this priming solution. We're going to add 800 microliters of the priming solution to the waste port to basically flush out that, kind of flush it out, the yellow liquid, which is storage solution. And see, so if you can see it, there's air right there. I'm going to push that out because we don't want air to be introduced. And I kind of dropped it on my list. There we go. We don't want to introduce an air bubble because we just sucked up an air bubble. Because if an air bubble gets in there, it will kill the pores. All I'm doing is slowly adding the 800 microliters of this solution into flow cell. You can see that. There we go. And now we let this sit for 5 minutes. So during this just this basically equilibrating right now. I'm going to set this back to 200 because we need to add another 200 microliters of priming solution after to get it at the correct volume. And oh, I forgot my timer. I'll be right back. All right, I'm back. Here we go. Timer's going. All right, so yeah, once this is done equilibrating, we're going to add 200 microliters of the rest of the priming solution to the waste port, and then we will add our library. Now, before we do that, we need to add the loading beads, which are right here. Mix it up. I'm gonna mix it up. Loading beads right here to our library that we made, and that's 25.5 microliters of the bead. I'm kind of doing this off camera because you see it's the loading beads are white. We're just going to add it to our library. There we go. And that's it. We just do a little tap. Set that back on ice, and we'll be ready to do that. And I'm going to change this to 75 microliters because that's how much of our library we're going to load. So that's how much volume is in that tube. And that's how much is going to go onto our flow cell. But it's only 250 nanograms of library that we're actually loading. The rest of it's the solution and the loading beads, and of course the library solution that would have been water, but we had to change it to that because that's what they said to use. So that's why. So it's doing its thing. Let me write down how much. Ooh, nope. There we go. I always like to write down what everything QC's at after I do it, so I'm gonna put it on here so I don't forget. And then we'll see sometimes, hopefully not, sometimes we lose pores on loading, which sucks. It happens. Not entirely sure why sometimes, but you can lose pores when you load your library. But yeah, so hopefully not. I've had that happen a few times, not recently, which is good. So we'll see. We will see. And then hopefully we'll start up sequencing. And then really hope that next time I do the sequencing, we'll be able to raid the actual sequencing channel, because then you guys can see the sequencing in real time. That'll be nice. It'll be a nice end for doing another sequencing run for us. Although there isn't any fun music to that, unfortunately. But that's okay. We're still figuring out how to do that. But you can watch the cool, it's like little squares that light up on this other big square. And you can watch as they turn different colors, meaning like, oh look, it's sequencing something. Oh look, that pore's dead. That kind of thing. So if you want it as a cool thing in the background, I guess. You can always have it a cool thing in the background. But we won't be able to do that today because it doesn't work. OBS does not work, unfortunately, due to an update. So, oh well. Oh well. Next time. Next time. Oh, we're almost ready. So about 30 seconds. And then we'll finish this up and get it going. And then if you have any questions for the end of the stream, let me know. I'll do just a brief overview of what we did today. And then I'll be ending stream after that. So it's about 4 hours. About 4 hours. I'm not too shabby. Like I said, on my own, just at the bench is about 3 hours for me. So it doesn't surprise me. I guess I figured out a good time for how to do it on stream. All right. So the first thing we do is we need to uncover this little port right here. You gotta be careful. So this is where the actual pores are. This is where we're gonna put our sample. Now we have to add 200 microliters of our priming solution that we made earlier. And remember, we don't want that bubble there. So we just carefully. Oopsie. Oh, well, it already went down there. It's all good. Gotta be careful. Do it even slower as it bubbles up into the. So this is the sample port. It'll bubble up into the sample port. I'm just trying to do it real slow. There we go. Okay. Now we add our library. I'm gonna mix it up a little bit. Of course, the loading beads slow down. We're gonna do 75 microliters. That's what we have for our library. And what we do is we just dropwise onto that, and it just capillary action goes right into it. And I'm a little shaky. I'm hungry. I'm ready for lunch. I am hungry, hungry. There we go. There, all right. Now we just put everything back. Move that back. And we close this. And the rest of it is just all computer stuff. Where I gotta name it. It's all in the computer at this point. So let me get this set up, and then I'm gonna move back over to the other spot, and then I will meet you there. So give me a couple minutes, and I'll be back with you. All right, of course. All right, just a second. Oh, I'm back over here. Let me fix the web camera. Let it think about its life again. There we go. There we go. All right, now we're back over here. We are back over here. It's looking a little... Just a second. Looking a little grainy. See if I can get the... Oh, that's fine. All right, just a second. I know why. I know why. It's not the way it's supposed to be. Because Logitech said no. Alright, just a second. Restart Logitech and hopefully it'll bring back my preset for it. There we go. Let's see. Did it bring back? Yeah, there we go. Much better. Yeah, every once in a while I gotta restart it, and then it's happier. All right. Well, there we go, guys. I hope you enjoyed the stream today. Let me do a quick recap of what we did. Let me bring back my notebook. I didn't turn my light on because it was a lot easier to see my notebook without that. So basically, what we did today was we did an enrichment, meaning we looked for a specific set of, or a specific sequence in the DNA, and then we used Cas9, which is the protein found in CRISPR. If you guys are familiar with CRISPR, it looked for that sequence. Where it found the sequence, it did a cut for us in the DNA, and when it did that, we needed to add an A using Taq polymerase, so an A, base pair A, to it, in order for the sequencing adapter, which has a T. Unfortunately, got a little smoosh. Let's see. Let me bring back my marker since the T kind of got. There we go. So in order to add this adapter to it, we needed to add the A using polymerase, which talked about as part of PCR. It's a protein that adds nucleotides to a sequence. Then we use a protein called ligase, which will basically attach DNA together. And because A pairs with T, we're able to ligate the adapter onto that cut sequence made by Cas9. And now we're sequencing it. So that's just a brand overview of what we did today for enrichment and sequencing. And we're doing long read sequencing, which means we're looking at long fragments of DNA. So in our lab, we've been able to sequence a fragment more than 200,000 base pairs long, so more than 200 kb, versus short read sequencing, where, like I said, I think Illumina's at 2,500, so very short fragments. So we're doing long read sequencing, so we're trying to get very long pieces of DNA. And that's so in the places that we are specifically targeting, we can see all the sequence around it, because we want to know what the sequence is. And with short reads, can't really do that easily. And that's because it'll chop it up and it'll be kind of hard with the downstream analysis, because the sequence we're actually looking at is very repetitive, so it has a lot of like AAA or TTT when I mean repetitive. So it's very hard in downstream analysis for it to do that. So if anybody has any questions, feel free to put them in chat. I'm so glad that you were able to be here with me today and go through the protocol. If you missed any part of this VOD today, it will be up on the YouTube channel later on. Let me put that in stream for anyone who might have missed it. So you can see it there. You can also see it at Twitch. It'll be up for, I'm not sure if they implemented it, but usually it's up for 14 days. It might be up for only 7 now. But you can go back and look at it. You can look at my other streams if you would like to on the YouTube channel. And If you need to know outside of Twitch when I'm going live, I post live messages on my Twitter, so you can follow me at Twitter. If you would like to follow me and know when the next time I'm going live on Twitch, obviously hit that follow button, hit that notification bell, and it will let you know when I'm going live next. But yeah, if there aren't any questions, I think I will end the stream here. It's been about 4 hours. So I'm glad for all of you who have stuck around for the entire time. And, well, I'm just glad everybody's here. I appreciate you being here, and I appreciate you enjoying the stream. So yeah. I'm just going to wait for just a second in case any questions come through. All right. Well, thank you so much for being here. I really appreciate it. Thanks for being at the bench with me today. And hopefully I will see you next time. And I hope you have an amazing rest of your day, night, morning, wherever you are. So thanks again. Bye.